by our eminent speaker, Dr. Suhasbhai Vyas Sahib from Bhakkavi Narsi Mehta University, Junagadh. He will deliver the lecture soon, but before the lecture, we have with us today our eminent guest, Miss Kanaklata, Dr. Kanaklata Madam. She is senior scientist and head of department in Krushi Vignan Kendra, District Panchmahal at Jalpur. And as usual, in our team, we have with us today's coordinator, our program coordinator, Professor Ajay Bhai Soni Sahib, and Life Science Coordinator Falguni Madam. Along with me, we have with us our two eminent student representatives, Saif Khan and Waghela Jaimi. So I heartily welcome all of you dignitaries present over here in the live session of 21st lecture. I also welcome all the students who are watching us live on YouTube and Facebook. So heartily welcome to all of you. To inaugurate the session, I would like to invite Professor Soni Sahib to initiate the lecture by providing best wishes to all of us and inaugurate the session. Soni Sahib, અવાજ આવે છે?
સર અવાજ આવે છે મારો? નાકર
જી
સાહેબ
સર, પ્લીઝ.
અવાજ?
યસ સર.
ઓકે. શ્રી ગોવિંદ ગુરુ યુનિવર્સિટીના ડિપાર્ટમેન્ટ ઓફ કોમર્સ દ્વારા આયોજિત SLET કોચિંગ યાત્રા એ છેલ્લા 3 મહિનાથી વધારે સમયથી ચાલી રહી છે. જ્યારે સમગ્ર દુનિયા COVID-19 મહામારીમાં ફસાઈ હતી અને એવા સમયમાં અમારા વીસી પ્રોફેસર પ્રતાપસિંહ ચૌહાણે અમને દિશા દર્શન કર્યું કે વિદ્યાર્થી માટે આપણે શું કરી શકીએ. વિદ્યાર્થી પાસે પૂરતો સમય ઘરે છે તો સમયનો સદુપયોગ થાય અને એમની ઉજ્જવળ કારકિર્દી બને તો એ માટે એમના દિશા સૂચન કર્યું અને એમને મને જવાબદારી આપી અને કીધું આ નેક્સ્ટ લેટનું કોચિંગ ચાલુ કરો અને આનંદ છે. શરૂઆતમાં જ્યારે એનું રજીસ્ટ્રેશન કર્યું ત્યારે માત્ર 563 વિદ્યાર્થી પંચમહાલ અને દાહોદમાંથી ભેગા થયા, પરંતુ 5 ફેકલ્ટીમાં આજે યાત્રા ચાલી છે. 3 મહિનાના અંતે હું કહી શકું કે લગભગ 7670 7,700 વિદ્યાર્થીઓ આ નેક્સ્ટ લેટ કોચિંગ દ્વારા ઓનલાઈન કોચિંગથી આપણી વચ્ચે જોડાયા છે. 5 ફેકલ્ટીમાં આપણો આ કાર્યક્રમ ચાલી રહ્યો છે. તેવા દિશા દર્શન કરનાર અમારા ફ્રેન્ડ ફિલોસોફર અને ગાઈડ વીસી સાહેબ પ્રોફેસર પ્રતાપસિંહ ચૌહાણ સાહેબ, અમારા ઇન્ચાર્જ રજિસ્ટ્રાર સતત પરિસ્થિતિ પોઝિટિવ અને ઉદ્યમી એવા ડોક્ટર અનિલ સોલંકી સાહેબ, શ્રી ગોવિંદ ગુરુ યુનિવર્સિટીના સૌ એસી મેમ્બર્સ, સાયન્સ કોલેજના આચાર્યશ્રીઓ, અધ્યાપકશ્રીઓ, વાલા વિદ્યાર્થી ભાઈઓ બહેનો, આજના વિદ્વાન વક્તા ફરીથી એકવાર અમારા કુટુંબના એવા નરસિંહ મહેતા યુનિવર્સિટીના આજના આપણા વિદ્વાન વક્તા રેસ્પિરેશન ઉપર આપણને વાત કરવા જેવા ડોક્ટર સુહાસભાઈ વ્યાસનું વેલકમ કરું છું. ગોવિંદ ગુરુ યુનિવર્સિટીના આખા પ્રોગ્રામ કોર્ડિનેટર તરીકે સુહાસભાઈ તો અમારા પરિવારના જ છે. ફરીથી એકવાર મને વેલકમ કરવાનો ખૂબ આનંદ છે એમને. આ ઉપરાંત આજે આપણી વચ્ચે ઉપસ્થિત છે કેવીકેના રિસર્ચ સાયન્ટિસ્ટ ડોક્ટર કનકલતાબેન, એ પણ આપણી વચ્ચે ઉપસ્થિત છે. નાકર સાહેબ અનેક નવા પ્રયોગો કરે છે. મને આનંદ છે કે સુહાસભાઈ 21મું વ્યાખ્યાન છે એકલા લાઈફ સાયન્સનું. આવા તો અમારી પાસે લાઇબ્રેરી સાયન્સના 10થી વ્યાખ્યાન, સંસ્કૃતના વ્યાખ્યાન, આખી સિરીઝ ચાલુ છે. આ મારું લગભગ 94મું વ્યાખ્યાન છે ટોટલ બધા ફેકલ્ટી થઈને. તો મને એનો આનંદ છે કે આ કરનારા ડોક્ટર રૂપેશ નાકર એ ગઈકાલે જ એ અમારી યુનિવર્સિટી સાયન્સના ગાઈડ બન્યા છે. તો નાકર ગાઈડ બનવા માટે પણ ખૂબ ખૂબ શુભેચ્છાઓ પાઠવું છું. અને એમની જોડે સતત ખભે ખભા મુલાવીને કામ કરનાર ફાલ્ગુનીબેન પરમાર, એ પણ અમારા સતત એક્ટિવ મેમ્બર છે. સુહાસભાઈ અને કનકલતાને જણાવું કે આપણી વચ્ચે વાઘેલા જયબીન અને સેફ ખાન પઠાણ 2 સ્ટુડન્ટ્સ પણ આપણી વચ્ચે ગુજરાતી ઉપસ્થિત છે. તો આજના વક્તા જુનાગઢથી છે, નર્મદ યુનિવર્સિટીમાંથી. અમારા કનકલતાબેન મુખ્ય મહેમાન છે. એ સાયન્ટિસ્ટ છે, એગ્રીકલ્ચર યુનિવર્સિટી માંથી છે. આ પ્રોગ્રામ થઈ રહ્યો છે ગોધરાથી, એ ગોવિંદ ગુરુ યુનિવર્સિટી છે. સાહેબ ગોધરાની સાયન્સ કોલેજ એસપીડી કોલેજ માંથી છે, અને ફાલ્ગુની બેન એ વાંટા વંશવડ કોલેજ માંથી છે. તો અને હું અત્યારે અમદાવાદ છું. તો આવું સૌનો સાથ વિકાસ એ આપણે પ્રધાન મોદી સાહેબની જે સંકલ્પ, એટલે આખા કાર્યક્રમના પણ પાર્ટિસિપેટ છે. ડાકર સાહેબ અને ફાલ્ગુની બેન પાર્ટિસિપેટ છે. રિસર્ચ સાયન્ટિસ્ટ બેન આપણી વચ્ચે ઉપસ્થિત છે, અને વક્તા જુનાગઢ થી નરસિંહ મહેતા યુનિવર્સિટી માંથી ઉપસ્થિત છે. તો મને ખૂબ આનંદ થાય છે. આ કાર્યક્રમ આપણે યુટ્યુબ અને ફેસબુક ઉપર લાઈવ જોઈ શકીએ છીએ. કોઈ કારણ આ કાર્યક્રમ ચૂકી જાય, આ કાર્યક્રમનું રેકોર્ડિંગ થઈ રહ્યું છે, પછી પણ જોઈ શકે છે. આ માટે મારી ટીમના સતત વેબએક્સ થી ઝૂમથી રવિવાર દિવસ હોય, 15મી ઓગસ્ટ હોય, તો પણ સતત સક્રિય એવા અમારા જાગૃતભાઈ પંડ્યા, મિલનભાઈ શાહ, એ સૌને પણ હું યાદ કરું છું, કેમ કે આના સહયોગથી આ કામ સરળ થઈ રહ્યું છે. 94 વ્યાખ્યાન હું સાક્ષી છું, પણ કામ તો મારી ટીમ મેમ્બરો કર્યું છે. ઝાઝા હાથ જાડિયામણા એ ન્યાયે સૌએ પોતાનો પૂરતો સમય આ નાકર સાહેબ અને ફાલ્ગુન માટે કહું એટલું ઓછું છે કે 14 15 કલાક પોતાના કોલેજનું કામ કરીને એક્સ્ટ્રા કામ કરી રહ્યા છે, અને એક પણ જાતના બીજાની અપેક્ષા વિના કામ કરી રહ્યું છે. એટલે એને હું સેવા યજ્ઞ કહું છું કે સેવા કરી રહ્યા છે, એમાં બધા આહુતિ છે. જેને જેને સમયનું દાન આપ્યું છે, પોતાના વિદ્વત્તાનું દાન આપ્યું છે, એ સૌ ઋષિ તુલ્ય માણસો છે. એ સૌના પ્રયત્નને હું બિરદાવું છું. ગોવિંદ ગુરુ યુનિવર્સિટી સુહાસભાઈને ફરીથી વેલકમ કરી રહી છે, કનકલતાબેન વેલકમ કરી રહી છે. ઓન બિહાફ ઓફ શ્રી ગોવિંદ ગુરુ યુનિવર્સિટી, I warmly welcome to my our chief of guest Kanaklata madam and our keynote speaker Dr. Suhas Vyas. Warmly welcome you on behalf of Govind Guru University પરિવાર. મને આનંદ છે કે અમે મોક ટેસ્ટ, હવે આ પરીક્ષા 24મી નેટની પરીક્ષા શરૂ થઈ રહી છે, તો વિદ્યાર્થી માટે અમે મોક ટેસ્ટ મૂકી દીધી છે. લાઈફ સાયન્સની 5મી ને આજે 6ઠ્ઠી મોક ટેસ્ટ મૂકવાની છે. ફાલ્ગુની મેડમે મને સૂચના આપી જેવી. તો આ મોક ટેસ્ટ દ્વારા પણ વિદ્યાર્થીના જ્ઞાનમાં વધારો થાય, અને પ્રેક્ટિસ મેક્સ પરફેક્ટ. અને મને આનંદ છે કે ગુજરાત સિવાયના બાકીના 7 સ્ટેટમાં પણ આપણી આ વીસી સાહેબ રહ્યા છે. તો સુહાસભાઈને પણ વિનંતી, ભારતમાં સંપર્કો છે, અને અમારી ટીમ પણ 7 સ્ટેટ પહોંચી ગઈ છે. અત્યારે વીસી સાહેબે સૂચના આપી છે કે આ કાર્યક્રમ આપણે ચાલુ જ રાખવાનો છે. હજુ સ્લેટની એક્ઝામ બાકી છે, પેટની એક્ઝામ બાકી છે, એમએસસી ની એક્ઝામ બાકી છે. એટલે આપણી આ વિદ્યાર્થી માટેની જ્ઞાન આપવાની પરંપરા અને આ યાત્રા ચાલુ જ રાખવાની છે. રોજે રોજ એમાં નવા નવા પીછા ઉમેરાય છે. ગઈકાલે રૂપેશ નાકર PhD ના ગાઈડ બન્યા. આ તબક્કે સૌ વિદ્યાર્થી મિત્રોને પણ એક અમારા મોટું પીછું કે ગોવિંદ ગુરુ યુનિવર્સિટીમાં લાઈફ સાયન્સના ગાઈડ ગોધરામાંથી ઉપલબ્ધ છે. ના કોર્સમાં અમે ગઈ વખતે 200 વિદ્યાર્થી રજીસ્ટર થયેલા, એમાંથી 100 ટોટલ 40 વિદ્યાર્થી શોધ અંતર્ગત વર્ષની 2 લાખ, એવા 2 વર્ષના 4 લાખ રૂપિયા કરતા કરતા લોકો ખર્ચ પેટે મળ્યા. 1 કરોડ ને 60 લાખ ગુજરાત સરકાર તરફથી અમારા વિદ્યાર્થીને મળ્યા છે. PhD કરનારા સ્ટુડન્ટ્સને આટલું મોટું સ્ટાઇપન્ડ, એને PhD ના કામમાં સરળતા રહે એ માટે ગુજરાત સરકારે સ્કીમમાં અમારા 40 વિદ્યાર્થીઓ સક્સેસ રહ્યા છે. તો આ સૌના પ્રયત્ન, વીસી સાહેબનું દિશાદર્શન, અને અમારા ઠાકર સાહેબ, ફાલ્ગુની મેડમ, અમારા રાજેશ કગરાના પાવી જેતપુરમાં એકલા આજે 35 સેમિનાર કરી રહ્યા છે. તો અમારા પાર્થ ભટ્ટ એ આખા ગુજરાતમાં ડંકો વગાડી રહ્યા છે. આ સૌના પ્રયત્નને હું યાદ કરું છું. સૌના પ્રયત્નોને બિરદાવું છું અને નાકર સાહેબ એની ટીમને ખૂબ ખૂબ જઈ રહ્યા છે. આઈએસબીઆરની બુકલેટ જે અમારા માટે એવિડન્સ બનવાનો છે. ગોવિંદ ગુરુ યુનિવર્સિટી આજે કોવિડ-19 ના સમયમાં ગુજરાતના ખૂણે ખૂણે અને ભારતના 7 રાજ્યો સુધી પહોંચી ગઈ છે. અમારી પાસે માત્ર 7 નો જ સ્ટાફ છે. આટલા ઓછા સ્ટાફમાં પણ અમારી પાસે 100 કરતા વધારે ડેડિકેટેડ અધ્યાપકો છે. 100 કરતા વધારે આચાર્યો અમારી જોડે છે અને અમારા 1 લાખ કરતા વધારે સ્ટુડન્ટ્સ અમારી જોડે છે. અમારી યુનિવર્સિટી સ્ટેટની 5મા નંબરની યુનિવર્સિટી છે કે 1 લાખ જેટલા સ્ટુડન્ટ છે. 150 કોલેજોમાં અમારું નેટવર્ક ગોઠવાયેલું છે. 5 જિલ્લા અમારા છે ગોવિંદ ગુરુ યુનિવર્સિટી અંતર્ગત. પંચમહાલ, દાહોદ, વડોદરા જિલ્લો, મહીસાગર અને છોટાઉદેપુર જિલ્લો. એમાં 3 જિલ્લા અમારા ટ્રાઇબલ બેલ્ટ છે. તેમે અમે આ કામ કરી રહ્યા છે એ અમારા માટે ગૌરવની વાત છે. જેને જેને અમારામાં સમય આપ્યો છે, વિદ્વત્તાનો આપ્યો છે, એ સૌને વંદન કરીને મારી વાણી વિરામ આપું છું. વંદે માતરમ. ભારત માતા કી જય. નાકર સર.
Ji, thank you so much, Soni sir, for your very kind and motivating words that definitely motivated all of us to work more and more. And we are thankful to you for such a guidance. Now, today we have with us our guest of honor, eminent guest, Dr. Kanaklata Madam. She is working as a head of department and senior scientist in Krushi Vignan Kendra of ICAR, Indian Council of Agriculture Research, Central Government, Government of India. And we are very happy to see you, ma'am, here. She is working for extension activities mainly and different experiments related with agriculture, specifically for farmers of Panchmahal and other areas of Gujarat. So we heartily welcome you, ma'am. And as you know that we are today organizing program for NET GSET preparation of students of Gujarat and other states. So we would like to have your best wishes and blessings. If you can hear me, madam, over to you. Kanaklata, madam.
વેલકમ.
बिल्कुल. हाँ, धन्यवाद सर. धन्यवाद रुपेश जी. एंड इट्स माय प्रिविलेज कि आपने मुझे आज ये मौका दिया कि मैं बच्चों से रूबरू हो सकूं, आप लोगों से मैं प्रत्यक्ष सामने होकर बात कर सकूं. अभी सोनी साहब जी कह रहे थे बहुत लोग बहुत जगह से जुड़े हुए हैं, तो ये बिल्कुल हमारे लिए एक सौभाग्य है कि इतने दूर दूर से सभी व्याख्याता सभी लोग जुड़ते हैं. तो मैं सभी लोगों का स्वागत करती हूँ. खास करके जो आज के जो इस लेक्चर सीरीज के जो मेन जो लेक्चर देने वाले आज हमारे जो विद्वान हैं उनका भी मैं स्वागत करती हूँ और साथ में प्रोफेसर साहब आपका भी करती हूँ, क्योंकि आपने जो अभी बातें बताई हैं रियली बहुत ही मोटिवेशनल है. तो आज मैं यहाँ पे हूँ तो मैं रुपेश जी का विशेष तौर पे आभार करूंगी, क्योंकि उन्होंने मुझे आज थोड़ी देर पहले ही बताया. तो मैंने बोला ये कोविड ने बहुत कुछ हमसे लिया है, तो कुछ अवसर भी हमें मिले, क्योंकि पहले हमें सोचना पड़ता था कि हम कहाँ नहीं है. लेकिन ये जो वर्चुअल मीटिंग्स जो इस कोविड के कारण ये ज्यादा प्रसिद्ध हो गया, इससे एक फायदा हो रहा है कि हमें दूर दूर के लोगों से बड़ा सहजता से हम संपर्क कर ले रहे हैं. आज जो विषय है वो तो मैंने सुना है सिस्टम पे आज बातें होंगी, लेकिन जो बच्चे आज भाग ले रहे हैं मैं उनको बहुत सारी शुभकामना दे रही हूँ कि आप आगे बढ़ें और आप अपने जीवन में वो मुकाम हासिल करें जिसके लिए आपने लक्ष्य निर्धारित किया है. दूसरी बात है कि ये सुन के मुझे बहुत अच्छा लगा कि गोविंद गुरु महाविद्यालय एक सीरीज शुरू किया है और 94 सीरीज है और जल्दी ही आप शतक बनाने वाले हैं. तो ये बहुत ही सराहनीय काम है कि ऐसे में लोग चुपचाप बैठे नहीं रहे. हमने एक रास्ता ढूंढा कि किस तरह से हमें अपने स्टूडेंट्स के साथ अपने लोगों के साथ में काम करते रहना है. तो कहते हैं ना कि हमें कोई हरा नहीं सकता है, मानव जब जोर लगाता है पत्थर पानी हो जाता है। तो हमने वो जोर लगाया है और ये कोविड से भी हम संघर्ष करते हुए ये काम को हमने जारी रखा है और इसमें गुरु महाविद्यालय का ये कदम बहुत ही सराहनीय है। मैं आप सब लोगों का दिल से आभार व्यक्त करती हूँ कि आप भारत के जो भावी कर्णधार हैं, जो आने वाले नागरिक हैं, उनके लिए आपने इतनी मेहनत की है। और आज 94 जो सीरीज है, मैं भी सौभाग्यशाली हूँ कि मुझे मौका मिला है। मैं बच्चों से सिर्फ यही कहना चाहूंगी, 2 बच्चे जो हमारे साथ में हैं, सैफ और बारिया कोई है ना, मैं नाम भूल गई दोनों बच्चों की। तो दोनों बच्चे तो रिप्रेजेंटेटिव नहीं है, लेकिन बहुत सारे बच्चे यूट्यूब पे, हाँ जी सैफ खान,
यस मैम,
जी जी।
यस
तो
मैम,
हाँ, आपके साथ एक और भी मित्र है ना?
यस मैम,
हाँ, जैमिन, हाँ जी, वाघेला
यस मैम,
जैमिन, हाँ। तो आप दोनों तो प्रतिनिधि हैं, लेकिन आपके साथ मैंने सुना है कि बहुत सारे लोग यूट्यूब पर भी लाइव आपको देख रहे होंगे। तो कितनी सराहनीय बात है कि इस तरीके से एक हमने टेक्नोलॉजी का उपयोग किया, क्योंकि हमारे प्रधानमंत्री भी कहते हैं कि टेक्नोलॉजी को सीखिए, बहुत जरूरी टेक्नोलॉजी को, तभी हमारा देश आगे बढ़ेगा। हमारे देश का पीछे रहने का एक यही कारण है कि हम नई तकनीकियों पे जल्दी से विश्वास नहीं करते हैं और उसमें हम आगे पीछे रह जाते हैं। मैं इसलिए भी ये बात कह रही हूँ क्योंकि मैं कृषि से संबंधित हूँ और कृषि के क्षेत्र में हमें किसानों के साथ काम करना पड़ता है। तो वहां पे बहुत ही मुश्किलात आती है कि जब हम किसानों के साथ में काम करने जाते हैं तो उनको टेक्नोलॉजी का पता नहीं होता है। अभी हमारे पास में मौसम विज्ञान का भी हमारे यहाँ काम होता है। तो मैं अपना अनुभव आपसे शेयर कर रही हूँ कि मौसम विज्ञान का भी काम होता है। उसमें मेघदूत ऐप है, दामिनी ऐप है, जिसमें दामिनी में जो बिजली, आकाशीय बिजली जो गिरती है, इसका हम तुरंत उनको बताते हैं कि इस क्षेत्र में यहाँ पे इतने मिनट के अंदर में आकाशीय बिजली गिरने वाली है। इसमें चल रहा है, अपने पंचमहल कृषि विज्ञान केंद्र भी है। लेकिन अब बात आती है कि ये टेक्नोलॉजी का उपयोग कौन कर सकता है? जिनके पास में वो साधन है तो उसका उपयोग करने का उनको तरीका। तो अब जो आप बच्चे नेट और GSET की तैयारी कर रहे हैं, तो आप तैयारी करेंगे और आप कहीं ना कहीं काम में जाएंगे। तो मेरा आप लोगों से एक यही निवेदन रहे, एक सबको तभी हम सैनिक कहलाएंगे। अगर हम अपनी तत्परता से निभाते हैं तो भी हम देश के सैनिक हैं और हम देश के लिए लड़ रहे हैं। की वजह से हम इतने पिछड़ रहे हैं। हमारे देश के पास संस्कृति नहीं है और दिमाग की कमी नहीं है। हमारे देश के जो प्रबुद्ध लोग हैं जाकर के इतना बहुत बड़े बड़े काम कर रहे हैं, लेकिन हमारे यहाँ वो काम नहीं हो पा रहा है। पर अब ये कमांड आज के बच्चों के हाथ में है कि वो कैसे करें। तो अपने काम को हमेशा बहुत ही तत्परता से करना चाहिए, सोच समझ के करना चाहिए और हमेशा सिर्फ अपना अर्थ नहीं बल्कि परमार्थ भी देखना चाहिए। अगर हम समाज के लिए जिएंगे तभी हम अच्छा जिएंगे। रुपेश जी से तो मेरी इस बात पे कुछ कहना ही नहीं है, क्योंकि जब वो शिविर लगाते हैं तो मैं देखती हूँ कि उस करवाते हैं, गांव के बीच में रहना, खाना बनाना। तो ये जो आप सीख रहे हो ये आपके लिए बहुत ही जिंदगी का एक रास्ता बना रहा है। तो रास्ता शुरू में थोड़ा कठिन लगता है, लेकिन बाद में इसमें बहुत बहुत ये सरल होता है, क्योंकि जब लोग आपसे सम्मिलित होंगे, जब लोग आपके साथ आएंगे तो आपको लगेगा काम। तो इस तरीके से आज का जो ये लेक्चर का जो व्याख्यान की जो कड़ी चल रही है, इस कड़ी की सफलता के लिए भी मैं कामनाएं देती हूँ कि रुपेश जी और गोविंद गुरु महाविद्यालय ने जो एक रचनात्मक कदम उठाया है ये सक्सेस हो और ये कड़ी बहुत लंबी चले और मुझे फिर से सौभाग्य मिले कि मैं फिर से आपके सामने आ सकूं और संभवतः मैं कभी अपने विषय पे आपसे बात करने के लिए भी आ सकूं तो मुझे बहुत अच्छा लगेगा। तो अब ज्यादा बात करना शायद यहाँ पे मुनासिब नहीं होगा, क्योंकि अभी आप जो सुनना चाहते हैं वो बहुत महत्वपूर्ण है, बहुत ही महत्वपूर्ण है। तो बातें तो बहुत सारी होती हैं कहने के लिए, लेकिन मैं सिर्फ इतना ही कहना चाहूंगी कि हर बुनियादी का एक ईंट है। तो इन बच्चों को हमें बिल्कुल बुद्धि से, ताकत से, सामाजिक रूप से मैं बल देती हूँ, क्योंकि आजकल मैं देख रही हूँ कि बच्चे मोबाइल में गुम हो गए हैं, इलेक्ट्रॉनिक गैजेट में गुम हो गए हैं। टेक्नोलॉजी सीखनी चाहिए, लेकिन टेक्नोलॉजी के नहीं हो जाना चाहिए, गुलाम नहीं हो जाना। ये मोबाइल आज कितना काम का है, जिसपे आप ज्ञान की बात सीख रहे हैं, लेकिन रात दिन आजकल पेरेंट्स की परेशानी है, बच्चों का अधिकतर समय मोबाइल, सामाजिक भूलते जा रहे हैं। तो आप समाज से जुड़े रहना, तभी आप भी सीखोगे जो का कोई फर्क नहीं पड़ता। तो मैं इन सब बातों के साथ में एक बात और भी कहना चाहूंगी कि बच्चों, आप लोग जो अभी संक्रमण का, कोविड का समय चल रहा है, मैंने जो अनुभव किया है, इससे लड़ने के लिए सिर्फ इम्यून पावर को स्ट्रॉन्ग करना। इम्यून पावर स्ट्रॉन्ग के लिए आप लोगों को आहार खाना बहुत ही जरूरी है। और सितंबर महीना पोषण पूरे हमारे की ओर से आप लोगों से मेरा है कि अपने इम्यून पावर बढ़ाने के लिए आप अपने भोजन का ध्यान रखें, क्योंकि जब तक इम्यून नहीं बढ़ेगा, आप बीमारियों से नहीं लड़ पाएंगे, आपका शरीर स्वस्थ नहीं होगा, तो आप हेल्दी रहना। और एक और भी छोटा सा निवेदन है कि अगर आपके पास थोड़ी सी जगह है कि आप अपने के पास में कुछ पौधे लगाएं। और मैं पौधों में बोलूंगी,
जी मैडम, आवाज आ रही है, प्लीज।
हाँ हाँ, कि पौधों में मैं चाहूंगी कि आप एक सहजन, ड्रम स्टिक, गुजराती में कहते हैं, जरूर लगाइए। एक मीठा नीम जरूर लगाइए, क्योंकि ये सिर्फ पौधा नहीं है, एक औषधि है। तो मैं ये भी आप लोगों से निवेदन भी आप जरूर करें। पढ़ाई के साथ साथ थोड़ा एक अपने एनवायरनमेंट के बारे में भी सोचना है। तो आज के इस कार्यक्रम में मैं गोविंद गुरु महाविद्यालय को बधाई तो देती ही हूँ और बहुत सारा शुभकामना देती हूँ कि वो बहुत अच्छा काम कर रहे हैं और आगे भी करते रहें। और उन सभी बच्चों को मैं आशीष देती हूँ और शुभकामना देती हूँ कि आप अपने लक्ष्य में सफल हो और आप अपनी जो तैयारी कर रहे हो इसको अच्छे तरीके से करो और आप अपना लक्ष्य प्राप्त करें। मुझे मौका देने के लिए बहुत बहुत धन्यवाद
जी, थैंक यू सो मच कनकलता मैडम, एंड definitely your best wishes are going to be fruitful to all of us and all our team members who are working for success of such kind of program, and definitely uh, it will be fruitful to all the students who are listening us. Thank you for giving your very precious time to us, Kanakalata madam, and we would definitely invite you again in the some of the lecture, as not only as a guest but as a lecturer also. So to continue the program, today we have with us our eminent speaker. जी सर.
આપણે ટાઈઅપ કરવાનું છે, એમઓયુ કરી દેજો તમે તમારી રીતે. આપણે
જી સર, અમારી વાત ચાલી રહી છે કનકલતા મેડમ જોડે ઓલરેડી.
હા, સરસ.
હે ને?
અને પાછળ બી मैडम थैंक यू. आपके साथ
धन्यवाद आप
अच्छा
लोगों
करना
का।
है.
बिल्कुल बिल्कुल
जी, Thank you, Soni sir and Kanaklata madam, and definitely we would invite madam to go for MAU with our university, so we can work together. And our vice chancellor always believes that work together and grow together. This should be the scheme of success of any of the organization or any of the city or area. So definitely Kanaklata madam is going to be there for us whenever required. Thank you again, ma'am, for your kind blessings. And today.
प्लेजर मेरा सौभाग्य थैंक यू
Thank you, madam. Today we have with us our eminent speaker, Dr. Suhasbhai Vyas Saheb. I would like to give his very short biodata, although his biodata is very strong. Dr. Suhas Vyas has more than 10 years of teaching and research experience. He is currently working as a head of Department of Life Sciences in Bhakt Kavi Narsi Mehta University. His area of expertise include drought and salinity tolerance in addition to environmental physiology. Earlier, he has worked as a assistant professor in Kutch University, Bhuj, and he was involved in one of the project of salinity tolerance there. And currently, he is also involved in one of the project funded by Department of Science and Technology, SERV. So he is very eminent personality in the field of ecophysiology as well. And today he is going to deliver lecture on the very important topic respiration. And definitely some questions are there in the examination, whatever the examination is, whether it is NET or GSET, GET or something like that. So we are very lucky to have you, sir, with us. And this is the second lecture of Suhas, sir. So we heartily welcome you, sir. And without delaying the time, I would like to invite Suhas, sir, to initiate the lecture on respiration. Suhas bhai, over to you, sir.
Yes, thank you very much, the whole organizing team. Thank you, sir. Thank you, Ajay bhai. Thank you, heartily thank you, and I first of all congratulate to be having 94th lecture of the series of this particular important aspect of NET as well as SLET, which the student demands. But unfortunately, in this COVID position, they were not able to go to their physical classes of the NET and SLET. But you people have made it, and it's hats off to you that you have reached up to 94th lecture of different types of topics of NET, for especially for NET as well as SLET. So respected Vice Chancellor Pratap Singh Chauhan sir, Ajay Bhai Soni, as well as Registrar of the University, Rupesh Bhai Nakar, Falguni Ben, who have managed to go through these such a long series of lectures, not, with the keeping the students in center in this COVID position. So, and also I congratulate Rupesh Bhai and other faculty members who have received their PhD guideship in particular in the subject of botany. Right, so congratulate, because now that university is growing in the research field also. I'm believing that, and I'm wishing that it will be in a very much fruitful topics which you will be giving towards the students. So straight away I will continue with my topic which I left in the last lecture. Just to give a short introduction about the lecture which I had taken in my last session, that was on photosynthesis as well as respiration. This part will be covering more on respiration, and I will be also talking on other aspect of the whole life science subject, that is about the cell biology, especially the cancer. So 2 points today I will be trying to cover as much I can in this scheduled time. So first of all, I will take with the process of plant respiration, through which last time in the last session which I had discontinued. So I am sharing my slides. So is the slide visible?
Yes, sir.
Thank you very much. Thank you. So today the lecture be, as I discussed earlier, that I have completed the part of the photosynthesis, which is the basic physiological aspects of the plant sciences. And today I will be covering on the second important aspect of plant physiology, that is respiration. So we all know that plant which produces the food with the help of the process photosynthesis, that is nothing but the glucose which is being produced as a product of as a food material in the plant cells, that is especially which is conducted by the leaves, which we had completed in the last session. And that food or that material which is to be utilized, which is to be broken down, that part will be covered by different metabolic activities. These activities, with out of these activities which is conducted for the survival of the plant, that is the respiration. So we need to have a closer look on the respiratory process which is going on in the plant cells. If you talk about, I have completed some of the part of this particular topic also in the last session. Just for a revision, I will be conducting this. If you see the respiration part, if there are three basic distribution or basic steps which can be included in the process, in the whole process of respiration, that starts with the glycolysis, maybe aerobic respiration or maybe an anaerobic respiration. The glycolysis, which we mean that the breakdown of the glucose molecule, which is to be compulsorily done by the cells, right? So whatever the system may be, that it may be aerobic, that may be anaerobic, but the plant have to go through the process of glycolysis. Now this glycolysis, if we dissociate the term glycolysis, that is the breakdown of the glucose. Glucose, lysis means breakdown. So that is a pathway through which the process of breaking of the glucose molecule starts. So we need to know about this glycolysis in a very quick time. We'll be seeing that. The second will be the Krebs cycle. The ATP through which the plant performs activities that are to be generated. Glycolysis will be breaking of the glucose molecule, producing a specific product out of through which the Krebs cycle, which will be utilizing that product for its own processing as well as producing the ATP, that is the currency of the energy. And finally, the third important step, that is the electron transport chain, or which is also being referred as electron transport system. So if you see the electron transport chain, there are various literatures if you survey, but if you refer to about the respiration or any of the other metabolic activities, the fourth one is oxidative photophosphorylation. Oxidative photophosphorylation as well as electron transport chain, they both are sometimes considered to be equal. Now I'll explain in my these slides how this mechanism happens for ATC as well as oxidative. What is the difference between these two? In my coming slides, I will be showing you in the particular presentation. Now, in general, if you see the respiration process, we know that the process is the release of energy. The respiration process leads to the release of energy. Now, this release of energy comes from a particular product. Now, that product is nothing but the food which is being prepared by the photosynthetic process. Now, this food is required to be broken down with the help of enzymes, with the help of other oxygen molecule. Now, with the food molecule of the oxygen as well as the enzymes, they constitute and they help in release of the energy along with the water molecule as well as carbon dioxide. So this is a general equation which we all have learned since our school days about the process of respiration as well as other part was the making of the food here in terms of photosynthesis. So if you consider the whole respiratory processes, we all are familiar with the terms aerobic respiration as well as anaerobic respiration. Now, just to show you the glimpse of these anaerobic as well as aerobic respiration, the slide which says that glycolysis, which is nothing but producing two molecules of the pyruvate from one single glucose molecule. So we have one glucose molecule, and always we have pyruvate produced from this one single glucose molecule. This pyruvate will be further divided or will be further divided into different processes. Say, for example, fermentation process, which leads to the formation of methyl alcohol as well as carbon dioxide, which can be released. And the second one, that is the muscle cells. We always find something that is a muscle fatigue, what we call it as. So this muscle fatigue is also due to the anaerobic respiration. oxygen in the cells, and which leads to the lactic acid. And the other important one, which we are going in very much detailed about in today's session, will be talking about the aerobic respiration, that refers to the different steps for the process of respiration. That the commonly, that is, as I told you earlier, that is glycolysis, Krebs cycle, etc., as well as the oxidative phosphorylation, which runs in ETC. I have prepared the slides in combination with the ETC. So glycolysis happens, or the location in which it is performing the mechanism, that is in the cytoplasm, that is outside the nucleus, or outside any of the organelle. So that is in the cytoplasm. Next one, that will be the Krebs cycle in chain, that will be running in the mitochondria. So you just imagine there are some of the enzymes which are present in the cytoplasm, but which are not present in the mitochondria. Hence, the cell have to perform the process of breaking down of the glucose molecule that is outside the mitochondria, and the product which is built in the cytoplasm has to brought in towards the mitochondria. So that will be some of the mechanism which we are going to understand, or which we are going to learn from this aerobic respiration. Then when we have reached towards the mechanism of the mitochondria, these mitochondria, as we all consider to be the powerhouse of the cell, the most common term which we have referred as a synonym, we can say, that is the mitochondria, which is also called as powerhouse of the cell. Now, why it is called as a powerhouse of the cell? As it generates more and more amount of ATPs, or more molecules of the ATPs are easily synthesized in this mitochondria. This is being in continuation the membrane of mitochondria, especially the inner membrane of the mitochondria. So the glycolysis produces a product supplied towards the mitochondria. Mitochondria has to generate brain. Now, there are some stages where the ETC works in a specific sequence, segments in the complexes of the whole ETC, that is electron transport chain. Now, as soon as it reaches, electrons are transferred, as the word itself suggests, that the ETC, electron transport, there is a mechanism of transporting the electron. So the term this ETC is restricted towards just the transfer of the electrons, only the transfer of electrons from one location towards the other, or maybe from one complex towards the other complex, then towards the other complex. And when there is a synthesis of the ATP, which is the major requirement of for the mitochondria, this ATP is being synthesized in this oxidative phosphorylation whole, as a whole process. It is also sometimes referred as Kemi Osmosis. Kemi Osmosis. So ETC in combination with Kemi Osmosis is referred as oxidative phosphorylation. So we need to understand the ETC part as well as the Kemi Osmosis part to understand the oxidative phosphorylation. So in sequence, if you see, the aerobic respiration starts with the glycolysis. As the glycolysis is being anaerobic, two pyruvate produced from one glucose molecule. These pyruvate molecules enters into the system, that is the for production of the ATP molecules through Krebs cycle as well as through the ETC, that is electron transport chain. So as I discussed earlier also, this particular slide, anaerobic as well as aerobic. Aerobic refers into the mitochondria for generating more number of ATPs, and these ATPs are in aerobic condition. ATPs can be produced in more numbers through aerobic respiration as well as through ETC. Anaerobic conditions, we can see that there is alcohol as well as lactic acid which can be produced. They also works in the cytoplasm. Here is to understand is the glucose molecule which is broken down, which can be broken down into pyruvate as well as two molecules of the pyruvate from one single. glucose. So if you see there, the glycolysis, production of pyruvate, just for that particular production of pyruvate enters into the Krebs cycle. This Krebs cycle is performed in the mitochondria, and we all know that mitochondria possess two membranes, that is inner membrane as well as the outer membrane. So it will be not that much easy for the pyruvate molecule to directly enter into the Krebs cycle. So there will be some mechanism for entering the pyruvate molecule or its other form for the mechanism for the starting of the Krebs cycle. And this Krebs cycle will continue with the ETC, that is electron transport chain, carried towards different complexes and finally producing the ATP. And finally producing the ATP through the oxidative phosphorylation. As I told you, this electron transport chain goes for the oxidative phosphorylation in combination, and this production of ATP can also be sometimes referred as Kemi Osmosis, and ETC as well as this Kemi Osmosis is referred as oxidative. Now, as I in the respiration will be for the glycolysis, maybe for the aerobic respiration or maybe for the anaerobic respiration. If you see this particular glycolysis in detail, we have two different phases in the glycolysis process. Two phases. We have, that is, the first phase is the energy where it is consumed, or energy which is utilized in the first phase is called as the investment phase. And the second point, or the second phase, second step, we can say, this second phase includes the production of the energy. The energy which is utilized in the first phase is also to be taken back, that is, the energy is produced in the second phase. We may have come across towards the different in lower standards also this process of glycolysis, and I also made aware about in my last session for the glycolysis. So I'll be quickly going through this particular process of glycolysis because I'm very much interested in showing the new topics for say about ETC. So the glycolysis, as I told you, that is breaking of the glucose molecule. This glucose will be broken down into two pyruvate molecule. We know that six glucose, that is 6 carbon, which are present in one single glucose molecule, are having the production of 3 carbon, that is 2 pyruvate molecules. Hence the equation is easily being balanced. And along with this 2 pyruvate molecules, we are also in search of energy production, and this energy production is nothing but the ATP. ATP is the energy currency where the cells can easily be utilized. So ATP, which we are referring to, as well as the different forms of energy molecules, maybe NADP, maybe FADP, FADH, all these are the energy-rich molecules. At the end of this particular slide sessions, we will be having an energy calculation that how much energy is produced. So the first phase is the investment phase. The second phase is the energy which we are producing from the glycolysis process. The glycolysis reaction, the first phase, includes the glucose molecule, and this glucose molecule, with the help of the hexokinase, the enzyme hexokinase, hexo, we all know that the hexo stands for 6. This hexokinase has the element that is kinase. Now kinase is meant for phosphorylation reactions. It is a thumb rule that wherever we see any kinase and any enzyme which is associated with the kinase, it has the efficiency of phosphorylating. Now this, as the word itself suggests, phosphorylating, that is phosphate is related with it. Hence here there is a transformation of ATP into ADP, right? So it is an important and crucial step where the kinase is involved. Hence hexokinase to remember, and the first step, that is glucose 6 phosphate, is being produced. Now glucose 6 phosphate can be isomerized, and with the help of the enzyme isomerase, this glucose 6 phosphate is changed into fructose 6 phosphate with the help of the enzyme isomerase. Fructose 6-phosphate once again has an enzyme which is very much actively associated with the fructose 6-phosphate and react with this compound fructose 6-phosphate, that is once again a kinase. So kinase related with the phosphate, addition or deletion of the phosphate molecule, obviously here there will be an addition of this fructose 1,6 biphosphate which is produced from the ATP. As I told you, the first phase is an investment phase, hence the energy is being utilized. So this energy is utilized, and this energy coming from the ATP molecules. These ATP is converted into ADP, hence one phosphate is being dissociated and entering into the fructose 6-phosphate is now converted into fructose 1,6 bisphosphate, or you can also tell is a biphosphate or diphosphate. So there are two phosphate molecules which are produced from one single phosphate molecule of fructose, and the enzyme which is interfering or which shows its interference, that is phosphofructokinase. Phosphofructokinase. Now this phosphate fructose 1,6 bisphosphate has a tendency to produce two different molecules. Now these two different molecules can interchange with each other. I'll show you in the later stages with the help of the enzyme aldolase. So this aldolase produces one of the aldehyde compound, that is glyceraldehyde 3-phosphate. It is most common and most familiar compound to all of us. This fructose 1,6 phosphate is no more its own compound. Now it is being dissociated into two important compounds, that is glyceraldehyde 3-phosphate and dihydroxyacetone phosphate with the help of enzyme aldolase. So this is the first phase. Now whatever energy which we have utilized in the second stage or in the second phase, we need to get it back. Now for that, this is the summarized form of the form. If you see, we have at two stages we have utilized the energy in form of ATP. Here, where the first was breaking of the glucose molecule and directly with the help of the kinase for glucose 6-phosphate. Next one, at the second stage, there was a kinase, and this kinase was also utilizing the phosphate molecule from the ATP and producing 1,6 bisphosphate. This bisphosphate produces two molecules, glyceraldehyde as well as dihydroxyacetone. These molecules, both these molecules, that is glyceraldehyde 3-phosphate as well as dihydroxyacetone phosphate, are able to convert itself or interchange itself at any time. So either the process can be carried out through this glyceraldehyde 3-phosphate, and as soon as it is completed, it can convert itself into once again this glyceraldehyde 3-phosphate from dihydroxyacetone. So this glyceraldehyde 3-phosphate further gives out an addition of the molecule from NAD and converts into NADH. Now, if you can recall, NADH is also energy-rich molecule. With the help of the enzyme GL3 phosphodehydrogenase, that is glucose 3 phosphodehydrogenase, and there is an addition of phosphate molecule over here. If you see, this is 1,3 bisphosphoglycerate, 1,3 bisphosphoglycerate. That once again here there is a bisphosphate, hence we have one more addition of the phosphate molecule. But the point to remember over here is there is a production of an energy rich molecule, that is NADH. At the end, it can be converted into ATP, that is NADH can be converted into ATP, a specific amount of ATP at the end of the reaction, that is at the end of the respiration cycle. So further, if you go, this 1,3 bisphosphoglycerate is converted into 3-phosphoglycerate with the help of the enzyme phosphoglycerate kinase. Now, as I told you earlier, once again there is an involvement of the element. So what will be there? There will be a phosphate addition or subtraction. Yes, here there will be addition, because we are talking about the second phase. This second phase is the gaining phase. Now we are not investing something, we are gaining out of our investment. So this kinase will help in producing the ATP molecules from the ADP by the phosphoglycerate kinase. And we have a compound that is 3-phosphoglycerate. 3-phosphoglycerate. This 3-phosphoglycerate can easily rearrange itself for further reaction. And this enzyme which helps in this whole process is the phosphoglycerate mutase. As the word itself suggests, there is an enzyme which is called which is associated with the mutase, that is just rearranging itself. 3-phosphoglycerate converted into 2-phosphoglycerate rearrangement of the phosphate molecules. And this phosphomutase further, that is 2-phosphoglycerate, further helps in production of phosphoenol pyruvate. Now we are very much closer to our required or expected compound, that is the pyruvate molecule, with the help of the enzyme anolase as well as an ion, that is Mg. This will help in production of the pyruvate molecules, that is phosphoenol pyruvate. And finally, in the last phase, in the last step, we can say, that is in the 10th step of the second phase, phosphoenol pyruvate once again gives a molecule which is required as an energy source, that is the ATP. This ATP is energy rich, and we are in this pyruvate molecule, which is further utilized in the Krebs cycle, as well as from the Krebs cycle, the energy which will be produced will be utilized for the ETC. Hence, phosphoenol pyruvate, with the help of the enzyme pyruvate kinase, will produce the compound, that is pyruvate. And here, of course, we are getting one more energy rich molecule. So if you see the overall second phase, there are 3 steps. At 3 steps, we are finding, or we are producing the energy rich molecules, that is through NADH, through ATP, from 3-phosphor during the production of 3-phosphoglycerate, as well as at the end step, that is in the 10th step, we are having a pyruvate molecule along with the ATP molecules. So in all, we have 3 steps which can produce the ATP molecules in the second phase. So the key points here over in the glycolysis cycle, that is 2 phases, investment phase as well as the gaining phase. In the investment phase, there were kinase enzymes which were producing from ATP to ADP. In the second phase, we are production of the ATP, that is the gaining phases. Hence, here we are producing ATP as well as NADH from. And also, the point here, that is only here in this particular session, there is only here glycerate phosphate is glyceraldehyde 3-phosphate is being included. We can also have similarly the production of dihydroxyacetane phosphate also. Dihydroxyacetane phosphate also. Hence, we have one pyruvate molecule from the glyceraldehyde 3-phosphate, and after the interconversion of dihydroxyacetane, we can have one more glyceraldehyde 3-phosphate which can produce another pyruvate molecule. That's the second one. So in short, we have one glucose molecule which is able to or efficient to produce 2 pyruvate molecules. Hence, the number of ATPs gained will be doubled in the second phase, obviously, because we have 2 molecules which can produce the pyruvate each. Right. So as this glycolysis, in the end of the glycolysis, we have seen that there are 2 phases. These 2 phases, the second phase is important one. Second phase has the ATP generation, that is gaining of the ATP molecules, which we have lost in the first phase. And the second phase important step was also to be noted that we have 2 pyruvate molecules, and these 2 pyruvate molecules are to forward. This forward process is the Krebs cycle. So the Krebs cycle starts with the glucose molecule, which has the production of pyruvate. This pyruvate now is to be utilized in the Krebs cycle. And this Krebs cycle, we are familiar that it generally doesn't take the pyruvate molecules directly. It has an intermediate compound which is very much familiar, that is acetyl coenzyme A, acetyl CoA. Now pyruvate molecule is a 3-carbon compound. Acetyl coenzyme A is a 2-carbon compound. See, I'm going a little bit of quicker in terms because I'm not taking the carbon molecule, number of carbon molecule for each and every compound. And you can understand, I've written in the slides that which can clearly mention the amount of carbon which is present in each compound. So pyruvate has 3 carbon, acetyl coenzyme has 2 carbon. Hence, there will be some release of carbon molecule from the pyruvate to produce the acetyl coenzyme A. This is done by pyruvate dehydrogenase, pyruvate dehydrogenase complex. So out of 3 carbon, we are having a compound which is a 2-carbon element. So 1 carbon dioxide is being released from the pyruvate, and also NADH is released during the process of production of acetyl coenzyme A. Now, this acetyl coenzyme A is entering into the Krebs cycle. Now the Krebs cycle starts. The step of the Krebs cycle starts. Now, as I referred earlier also in the earlier slides, glycolysis works in the cytoplasm. Krebs cycle has the pyruvate. Pyruvate is to be converted into acetyl coenzyme A, but this Krebs cycle works in the mitochondria. So the acetyl coenzyme A is being taken or brought into the mitochondria. So here in the mitochondria, what happens is the first step, which is in your slide, which is present in the slide. The first step involves the combination of 2-carbon compound, that is acetyl coenzyme A, in combination with the 4-carbon which is already present in the mitochondria, that is oxaloacetic, or we can also say it as oxaloacetate. So acetyl coenzyme A, 2-carbon, oxaloacetate, 4-carbon. Hence, we need a compound which will be having 6 carbon in one molecule. That is citrate or citrate. This citrate is having a 6 carbon, from 2 from acetyl coenzyme A and 4 from oxaloacetic acid. And the enzyme which works first reaction, that is citrate synthase. Synthase means, what is synthase? Synthase means production, to synthesize something. And here we are in search of a compound which can be a stable one, that is a citrate synthase. That is enzyme which works on for the production of citrate, that is citrate synthase. So the citrate compound, that is citrate having a 6-carbon molecule, and convert itself into a similar kind of compound, that is isocitrate with the help of the enzyme aconitase. Now here this aconitase is working for production of the isocitrate converts itself in the third step into alpha keto glutarate. But the point to remember over here, the key word is the release of carbon dioxide, and there is a production of NADH, which is considered to be the energy rich molecule. See, any cycle or any pathway which you are remembering, or which you are trying to remember, the key point to remember here is just understand the steps which involve the production of energy rich molecules. It will be very easy for you to understand why we are learning these processes, just to know about where the energy is being produced. And these are the steps which involve the production of the energy. Hence isocitrate in the third step, when it is converted into alpha keto glutarate, releases the carbon dioxide as well as it releases the NADH, that is the energy rich molecule, with the help of the enzyme dehydrogenase. So we started with a compound, that is citrate, that is having a 6 carbon. We had a isocitrate which is also having 6 carbon. Now we are removing one of the carbon molecule, right? One of the carbon molecule that is having a 5 carbon compound, that is alpha keto glutarate. Alpha keto glutarate, that is having a carbon which is released through the conversion of isocitrate into alpha keto glutarate. So similarly, in of the Krebs cycle, there will be a production of succinyl coenzyme A, which is also having 1 carbon less compared to that of the earlier step. So we have a 5 carbon in the alpha ketoglutarate. It has to release one more carbon dioxide to make itself stable. At the beginning of the lecture, as I told you, that the process of respiration involves the release of energy. Now this release of energy has different steps. The first step is the glycolysis. The second step is the Krebs cycle. Now here in this Krebs cycle, there is energy release. Now this energy release is nothing but these molecules, that is NADH. So alpha ketoglutarate, having the 5 carbon compound, is being now converted into a 4 carbon compound by the release of carbon dioxide in the fourth step, that is production of succinyl coenzyme A by the enzyme alpha ketoglutarate. Once again we have a dehydrogenase enzyme, hence there will be a production of NADH. So this succinyl coenzyme A will be having one more, it's similar type of product which is formed and which is also succinic acid, or succinate, the compound which is formed from succinyl coenzyme A in the fifth step with the production of one more energy-rich molecule, that is GTP. It is succinyl coenzyme A synthase, which works in production of succinate or succinic acid. So this succinyl coenzyme A synthase produces also this energy rich in the fifth step, that is G. So we have a citrate that is having converting itself into isocitrate once again converting into alpha ketoglutarate with the release of carbon dioxide. This alpha ketoglutarate is now converted into succinyl coenzyme A, once again release of carbon from its compound, and this succinate coenzyme A now produces and has become more little bit of stable compound which is called as succinate with the help of the enzyme synthase, that is succinyl coenzyme A synthase. This succinyl coenzyme A synthase is producing this GTP, which is very much similar towards the structures like ATP, NADH, or FADH, etc. So succinate now is converted into fumarate with the help of the enzyme succinate dehydrogenase. Now once again we have a hydrogen removal. Now here this hydrogen removal is nothing but ubiquinol is being released. Now ubiquinol is also a very good compound which has a tendency or efficiency to transfer the electrons, and these electrons are nothing but the energy rich molecules or energy rich forms. So ubiquinol is can be easily converted into FADH or FADH2. So ubiquinol, that is referred as QH, comes into the picture during the formation of succinate towards the fumarate, and this ubiquinol is also important in the ETC, that is electron transport chain. We will go in that particular slide. I will explain you how this ubiquinol is able to transfer the electrons. So as we have produced this fumarate in the sixth step, there is also a release of energy, that is in terms of FADH2. Compound, it has an enzyme which is the fumarase. This fumarase finally produces the malate, and this malate has been once again dehydrogenased by the enzyme malate dehydrogenase to produce NADH as well as oxalic acid once again. So as we know, it is referred as a cycle. So we need to produce the original compound or the previous compound with which we have started the reactions. So we had acetyl coenzyme A, which was helped by this oxaloacetate, which was already present in the mitochondria. This oxaloacetic acid or oxaloacetate will be formed once again from the melee in the 8th step. So we have a citrate produced from oxaloacetic acid and acetyl coenzyme A, 4 carbon, 2 carbon, having a 6 carbon compound. This 6 carbon compound at the second step forms alpha ketoglutarate, which is a 5 carbon. This 5 carbon is once again being broken down, and 1 carbon is released into succinyl coenzyme A towards succinate production of energies. Now, if you see the production of energies, we have a carbon released over here, we have a dehydrogenase, and we have NADH. You just focus on some of the NADH components over here. NADH, we have GTP, we have FADH, we have once again NADH in the 8th step. So all these steps are to be 100% memorized, because we are learning towards the production of energy, respiration, energy release. So this energy release is NADH, we have GTP, as well as we have FADH. So all these molecules are nothing but the currencies of the energies. ATP is considered to be the cash currency, the cash amount which can be utilized anytime, anywhere. NADH, we have FADH, we have GTPs. All these are the check currency. They are first of all, and then they can be utilized. Have energy from the Krebs cycle towards the process of respiration. Now, if you summarize the whole Krebs cycle, we have 3 NADH, ATP, as well as 2 carbon dioxide, which are produced through the Krebs cycle. Now, the production leads towards one glucose molecule. Now, as I told you, that glucose molecule forms pyruvate molecule, but there are 2 pyruvate molecules which are produced from one single glucose. So hence, glycolysis works subsequently for production of the pyruvate molecules. And each pyruvate can produce, here we can say the same cycle works over here also with the another pyruvate molecule. So it can be doubled. We have 6 NADH, we have 2 FADH2, we have 2 GTP, as well as we have 4 carbon dioxide, which can be released through the process of Krebs cycle. Hence the electron, which are in the electron transport chain, all these energy rich molecules are able to synthesize the ATP, that is the cash currency, as I told you earlier. So we have the energy rich molecules produced from the Krebs cycle, and from Krebs, the glycolysis was the pioneer one, which has converted glucose into pyruvate for the production of acetyl coenzyme A, and finally we have the energy rich molecule enter into the electron transport chain. Now here the point is, the NADH as well as FADH2 are considered to be one of the important energy rich molecules which helps powerfully in the electron transport chain. This electron transport chain works in the membrane of the mitochondria. Now, I'll not go into the detail of the different structures of the mitochondria because it takes time. So I'll just go towards the overview of the electron transport chain and overview of the mitochondria structure. If you see this particular slide, we can see there are different structures available on the internet also, but the main part is the inner mitochondrial membrane, the outer mitochondrial membrane, as well as the enzymes which are present inside the mitochondria. So we need to understand which are the enzymes. We need to understand where this inner mitochondrial membrane and how the electron are being transported in the inner mitochondrial membrane, as well as how these enzymes works on the transportation of the electrons. So if you see this particular slide, there are phases where I have shown you, that is segments. If you see the Roman letter which is written as 1, 2, 3 and 4, right, there are 4 different types of segments. We have started about the electron transport chain. So there are 4 different segments in which the whole electron transport chain can be divided. Now these 4 systems or 4 segments are referred as complexes. They are referred as complexes, and these complexes works individually. The point is very important. The complex 1 works individually, complex 2 works individually, 3 as well as 4. Now the whole complex chain where the electrons are transported from one towards the other, they works independently. One transport electron towards second, second towards the third, and third towards the fourth. So the system where the electrons are transported is called as electron transport chain. Now here in the slide, particular slide, the term is also where the ATP synthase is being written, it is also referred as chemo osmosis, that is Kemi Osmosis. Now this Kemi Osmosis is nothing but the composition, or what we call it as there will be a grade. I will show you as I continue my slides in the next slides. There is a variation in the chemical gradient, hence it is referred as chemical, because we know that if there is a balance in the solution which is present in the mitochondrial matrix or in the mitochondrial membrane, there will be an imbalance created during the electron transport chain, and due to which there will be a change or there will be a gradient developed, which is nothing but the chemical gradient or electron gradient, we can say, which is referred as chemiosmosis. The combination of electron transport chain as well as the with the chemiosmosis is referred to as oxidative phosphorylation. So in many literatures we can see that oxidative phosphorylation says same thing about the electron transport chain as well as the ATP synthesis, but if you consider only the electron transport chain, that says about the transportation of the electron from one complex towards the fourth complex only, where different types of segments are involved, different types of compounds are involved, and it has the efficiency of passing the electrons or transporting the electrons from one segment towards the fourth segment. Whereas in the chemiosmosis, it deals with only the production of ATP, nothing else, only the production of ATP. And why it does? It does due to the chemical gradient which has been developed due to the electron transport chain, and the combination is called as oxidative phosphorylation. So the first complex is referred as first complex, second, third, as well as the fourth. Now in this slide we have shown you about the different complexes which works with the electron transport chain, that is complex 1, complex 2, as well as complex 3 and 4. All these complexes works very much simultaneously. No complex overcrosses the other complex. That is the important point which is to be highlighted. That is, there will be no other electron which directly goes towards the second complex. So the initiation is to be done by the first complex only. Now this figure very much clearly says that there is an inner membrane space, there is an outer membrane, as well as there is an inner membrane of the mitochondria. So there are 3 segments, and on these 3 segments the whole electron transport chain works. So if you consider the first Krebs cycle, we have cleared the Krebs cycle, glycolysis as well as the Krebs cycle. So you just recall, in the Krebs cycle there were 2 important compounds which were produced. The first one is NADH and the second one is FADH, which was produced from the ubiquinol, right? So consider the NADH. So first complex, directly the NADH is involved. NADH cannot include or cannot go towards the second complex directly. There will be only the transition for the NADH to move towards the first complex only, and then the electron transport starts. Now we also will go in detail about what are the names of these complexes, that is from first towards the fourth. There is a designated name for each and every complex. But just to show you the overview about the electron transport chain, the first complex as well as has an association with the NADH. So FADH will not get involved in the reaction for the NADH as well as the first complex. So this is the mechanism where ATC starts. From the Krebs cycle, two compounds, NADH and FADH2, gets involved in the ETC, that is electron transport chain. So these electrons from the NADH move towards the first complex. This first complex is passing the electron from the first complex towards the fourth complex, So the chain works subsequently or consecutively. First, then second, then third, as well as then fourth. So there will be no overcrossing for each and every segments or each and every complexes. It has to move from one of the sequences. Now, another point which is at this particular time, which is to be remembered, that is, as the electron transfers from one complex towards the other complex, one important fact happens. Now, what is that? It is the protons, that is H plus, are always being shuttled from the inner mitochondrial intermembrane space. So whenever there is a transportation of electron, there will be the pumping of H plus ion. This pumping of H plus ion has the space which is called as intermembrane space. This H plus ion has the tendency to gather itself in this particular area or in that space. So we have the electron transport which is moving towards first, second, third to fourth, and during each time there will be the bombardment or pumping of the H plus from the matrix towards the intermembrane space. So this is the H plus ion gathering or H plus ion segregation towards the process, towards the intermembrane space. So if you consider the whole segments or whole complexes throughout this particular four complexes, at each and every time there will be H plus ion concentration which will be increasing in the intermembrane space. This will increase the balance of H plus ion in the intermembrane space. Hence, there will be some changes, that is more number of positive ions in the intermembrane space, and there will be a negative effect in the matrix, that is in the inner mitochondrial membrane, due to which there will be a gradient which will be taking place, and this gradient is called as electrochemical gradient. Hence we have referred to that particular term, that is Kemi osmosis, right? So what happens now, as the term osmosis is being referred, what happens is these elements or these ions, that is H ions, having a higher concentration towards the in the intermembrane space, are to be brought inside the matrix, or are to be brought or crossing the inner membrane space, inner membrane. But it is not that much easy. These H plus ion cannot move directly towards the inner membrane, because these inner membrane are very much not permeable, or we can say that is semi-permeable membranes. Hence, from the inner space towards the matrix. Hence, there should be some mechanism, and this mechanism is referred as another enzyme which helps is, that is ATP synthase. This ATP synthase helps in working off or bringing the H plus ion into the inner membrane. into the inner membrane, and this is called as Kemi Osmosis. The combination of this electron transport chain as well as this Kemi Osmosis, that is nothing but which is worked by the ATP synthase, that is bringing the H plus ion towards this area, and with the help of which ATP is being produced from ADP, right? So we have a ATP production in the mitochondria in more number. As and when H plus ions are brought inside, H plus ions are brought inside this particular area, that is in the inner membrane, they are obviously producing the ATP from the ADP, that is conversion of ADP into the ATP. So this is an overview of the whole electron transport chain, as well as we can say that is oxidative phosphorylation, which is referred as one of the steps in the cellular respiration, that is in the respiratory processes. So we have 4 different complexes which creates a electrochemical gradient in the intermembrane space, and this intermembrane space has more concentration of these H plus ions. These H plus ions cannot be brought inside. Why? Because of the semi-permeable property of the inner membrane, and this is done by the ATP synthase. ATP synthase produces the ATP and in the matrix of the mitochondria. Now, as I was referring, that was the overview of the electron transport chain. So we need to understand the names of these complexes. So for your information, I have kept the names of these different four major complexes which are involved in this electron transport chain. The first one is NADH dehydrogenase complex. NADH dehydrogenase complex. Now this dehydrogenase, as the word itself suggests, that is having dissociation of the water molecule, or we can say that is they're having a dehydrogenase complex. It is also referred as NADH oxidoreductase. NADH oxidoreductase. The peculiarity of this particular complex is they are having a L-shaped structure. This L-shaped structure, they are all nothing but the proteins and enzymes. So this is we can also say it as an inverted L-shaped structure, which is halfly embedded in the inner mitochondrial membrane, which is L-shaped structure. So it is called as NADH dehydrogenase complex. The word itself suggests that it converts the NADH as well as it is a complex. Hence, there may be some of the other elements also which are present along with the NADH. So it removes the hydrogen, that is dehydrogenase. It removes the hydrogen from the NADH along with the complex system in which it is present, that is obviously FMN and FES. Now FMN is flavin mononucleotide. Flavin mononucleotide, and FES is also always, that is iron sulfur complex. Iron sulfur complex, that is a combination of iron as well as it is a sulfur present over here. So the first complex name, that is NADH dehydrogenase complex, and it is removing the hydrogen from NADH. That is, hence if hydrogen is removed, obviously there will be a release of the electron. This electron, as it is released, it is being captured by this complex. As I told you, this electron is being released, and this electron, as it is released, is being captured by the first complex. The second complex is called as succinate dehydrogenase complex. Succinate dehydrogenase complex. Now, recently we have came across with this particular compound, that is succinate in the Krebs cycle. In the succinate, which is converted into fumarate by succinate dehydrogenase, it produces the fumarate in the Krebs cycle, and at that time also it was the releasing the energy, that is nothing but FADH2 with the help of the ubiquinol. If you recall, it is the ubiquinol which helps in the process, that is succinate dehydrogenase complex. This succinate dehydrogenase complex is called as a complex because it also has clusters of FeS, that is iron sulfur clusters. Hence it is called as complex. Succinate dehydrogenase is the second complex in the ETC. So this succinate dehydrogenase complex, that it was the second, moves the electron towards the third complex that is called as cytochrome reductase. That is cytochrome reductase. Now we all know that cytochrome are nothing but the group of proteins. It is also called as complex because there are iron cores which are present in this cytochrome reductase. These iron core has the tendency to convert from ferrous to ferric as well as from ferric to ferrous. So they interchange itself according to the concentration or according to the environment in which they are present. So they are called as cytochrome reductase, that is the third complex, which is one of the crucial complex for transportation of electrons. This third complex, as I told you, it is one of the crucial complex because it has different types of cytochrome present inside this complex. Along with the iron, and as I told you, iron has the tendency to convert itself into Fe2 to Fe3 and Fe3 to once again to Fe2. This cytochrome reductase has in itself cytochrome C1 as well as cytochrome D. Now, when the electron transport is transported into the third complex, it changes, it accepts the electron for cytochrome C1, and then it transports this cytochrome towards the cytochrome B complex. So, in all, the whole complex, that is complex 3, involves different types of cytochrome. Cytochrome C1, cytochrome B. Now, along with the cytochrome, there are also some of the other elements which are also present, and eventually this particular whole system is called as cytochrome reductase, or this complex is called as cytochrome reductase. Now, the fourth complex is called as cytochrome C oxidase. Cytochrome C oxidase, and this cytochrome C oxidase has different units which are of copper units. These copper units helps in transporting the electrons to make the electron more efficient for production of ATP or more production of ATP. That is the fourth complex, is cytochrome C oxidase. As soon as the third complex, cytochrome, the cytochrome reductase, this cytochrome reductase, that is cytochrome C1 to cytochrome B, then it transports the electron to cytochrome C, and eventually this cytochrome C passes the electron towards the fourth complex, that is cytochrome C oxidase. Now, why oxidase? Because this oxygen is now able to accept this electron and convert into the water molecule, that is H2O. The whole mechanism or whole sequence of electron transporting from complex 1, maybe NADH, complex 1 towards the complex 4 is acceptance of the electron, and here this electron is accepted by the oxygen to form the water molecule. The water molecule is, as soon as it is formed, there will be number of other variations which are happening in the whole area, that is in the mitochondrial matrix as well as in the intermembrane space. Now, as I told you, all these complexes are running individually. For example, flavin mononucleotide, as it receives the electron, this is being transported towards the this iron sulfur complex, and this iron sulfur complex passes the electron towards the ubiquinone, and ubiquinone, as it reaches towards the second complex, it helps in transporting the electrons towards the second complex. Ubiquinol is, as I told you, it is a freely permeable substances in this particular membrane. Hence, it moves the electron from one place to another place. And as all these electrons are transported, there will be the H plus ion, which will always being pumped into the intermembrane space, right? So as I have earlier also mentioned that I'm going to very much quicker in terms of this particular ETC. FMN transports the electron towards the ubiquinol. This ubiquinol has the tendency to accept those electrons, but in the second complex, there is also succinate which is present. This succinate dehydrogenase complex has the tendency to convert itself into fumarate, as we have seen in the Krebs cycle, and there will be FADH2 which will only combine with this second complex. Hence there will be addition of one electron from the complex 1 which is being transported to COQ, that is the second complex. Another electron which is also being received from FADH which is to be transported. Hence there will be 2 electrons from the first 2 complex itself. You just imagine the amount of H plus ion which is always being pumped into the complexes or into the intermembrane space at each and every time. So as this particular ubiquinol receives the electron, it transports the electron in a specific sequence in the third complex that is dealing with the cytochrome. This cytochrome C1 transports it towards the iron sulfate complex and to the cytochrome B complex, that is the whole third complex. This cytochrome B transports towards the main complex, that is cytochrome. The cytochrome molecule now is able to readily receive the electron in the third from the third complex, that is cytochrome. Now this time you just imagine one electron when it is transported in the third complex, it has the tendency to pump out 4H plus ions. So all these four, similarly as it is receiving FADH2, as it is receiving NADH, each and every time the electron is transported, H plus will be released. H plus will be accumulated in the intermembrane space. Now moving towards the complex number 4, the cytochrome C having the electron transports it in the fourth complex, which is a cytochrome oxidase, but it is also having the copper ions in it. These copper ions are also divided into subunits. For example, copper ion A type, copper ion B type. All these copper ions will be having a transportation of the electrons to generate more amount of ATPs, to generate have a efficient amount of ATPs. Hence this cytochrome behaves very important because there are more number of compounds where the electrons are to be transported. This electron are to be transported into cytochrome A as well as cytochrome A3, and then cytochrome A3, as it releases the electron, it is accepted by the water molecule. The water molecule, which is the oxygen molecule, and this is forming the water molecule. Hence, all these electrons, whenever they are transported, they increase their efficiency. Now, as H plus is being gathered at one place, that is, as I told you, that is in the intermembrane space, they will be pumped into one particular area. There will be more ionic gradient which is present. For that, there is no other option because the membrane itself here is impermeable membrane. This membrane will not allow the H plus ion to easily move. So what is the mechanism? That is ATP synthase. This ATP synthase has the conversion to form the ions which can be utilized for production of ATP. For production of ATP through ADP, that is H plus will be utilized by ADP and converted into the adenosine triphosphate, that is ATP, by the process what is referred as phosphorylation. So in short, this is the particular slide through which we can understand each and every step of electron transport chain. This electron transport chain starts with the mechanism of complex 1, complex 2, complex 3, 4. and up till here it is ETC, and here it is Kemi Osmosis, which is referred as, the whole combination is referred as the the oxidative phosphorylation. So if you see this this respiratory process starts with the glycolysis, and glycolysis runs with the runs for the production of pyruvate, which will help in Krebs cycle, and Krebs cycle eventually goes for the production of more number of ATP through this particular process, that is ETC, electron transport chain. Now if you conclude the whole respiratory mechanism of the cells, it is very interesting. If you see the three types or three pathways or cycles, we can say these three pathways generate energy, right? Consider glycolysis, Krebs cycle, as well as electron transport chain. If you see per glucose molecule in this respiratory system, this glucose molecules will be producing pyruvate. Pyruvate molecule during the production will produce two ATPs and two NADH, right? These two ATPs and NADH will be, this pyruvate molecule will be converted into acetyl coenzyme A with the production of, as you may be recalling, about carbon dioxide is being released and NADH is produced. So here also NADH is produced. So glucose molecule for the process of glycolysis produces two ATPs as well as two NADH for production of pyruvate. Once again, pyruvate is converted into acetyl coenzyme A for the two production of two NADH molecule. Now, if you see the Krebs cycle, similarly, there are two JTP molecules which are produced, 6 NADH are produced, as well as 2 FADH is produced, right? So this is the energy which is used from the glycolysis, and side, if you see the only electron transport chain. Now if you sum total of all these energy rich molecules, if obviously the electron transport chain has a tendency to have more amount of energy currency, more amount of these two NADH will be calculated, these two NADH will be calculated, 6 NADH as well as 2 FADH2. Each and every time 4 H plus ions gives us 1 ATP extra, right? Each and every 4 H plus ions will equivalent to 1 ATP. So all these all these electrons, whenever they are transported from one complex towards the other, each and every time 4H plus ions are pumped out. So we can say that more number of electron transport chain can be easily or we can say ATPs are being produced through electron transport chain, which occurs in this particular amount. 1 NADH equivalent to 10 H plus ions, which is equivalent to 2.5 ATP molecules. Similarly, 1 FADH2 gives us 1.5 ATP molecules. So likewise, if you can calculate all the ATP molecules produced from glucose, produced from Krebs cycle, as well as from this ETC. So the amount of energy which the cell, I'm talking about only one single mitochondria, right? There are different mitochondria working in the cell, and each and every mitochondria has the tendency to produce or to perform the Krebs cycle as well as to perform the electron transport chain with the help of oxidative phosphorylation. So as the whole oxidative phosphorylation. So oxidative phosphorylation brings out the whole energy bundle for the mitochondria, eventually for the cell to produce the energy, right? So we can say that there are 5 ATP produced from the during the acetyl coenzyme A, as well as we have total amount of 32 ATP molecules per glucose. So the whole process of this respiration starts with this particular glycolysis and ends towards the oxidative phosphorylation in the process of respiration. That is, we can call it as a cellular respiration at the end. right? So I'll be moving towards the next different topic, though I have only specific time in it, but I want this whole respiration in a very short time, but I could not finish it. But I'll try my best to start this topic of cancer, which I'm supposed to take it today. So I'll continue with that, and let's see if the time permits. Okay, so I'm hope I have is cleared.
Yes, sir.
Thank you for the
Okay.
response. Fine. So we will moving towards the another topic, which is from the life science syllabus, and that will be a little bit of different from the respiratory photosynthesis, but it is dealing with the today's issue, today's health issue, we can say, that is about the cancer. So it is a crucial point to discuss on this particular platform that how to understand cancer, what are the phases or what is the mechanism of the cancer, how to deal with the cancer. There are various aspects, there are various researches which are going on. Hence it is a compulsive part that the life science syllabus have to include the particular topic which deals with the current scenario of the cancer disease. And just for a glimpse, I have shown you this little slide. There will be no time that out of 3 patients, one will be dealing with the cancer. Not only one specific cancer, there are list of cancer which are being given by the United Nations as well as the World Health Organization. There are various types of cancers, but out of 3%, there will be one patient who will be dealing with the cancer disease. So it is a high time to understand this cancer and to know about its mechanism. And for that, to understand the cancer, we also need to understand our own immune system, how it is working. And to make powerful immune system, we need to take some of the precautions. As the current scenario says, the immune system, as it is powerful, you will be free from the corona, or you will not have to suffer from the disease that is COVID. So this immune system, how it works and how it affects the cancerous cells, and why it is not able to affect the other parts of our cells, we'll talk about this immune system, and as well as the important point that oncogenes. This will be the basic metabolic pathways which the cancer cells goes through their livelihood in our body. Some of the important points that is dealing with first. If you see in this particular slide, though I will be sharing the slides, and I also will be convinced towards organizers also. So you can see on the YouTube channel after the recording. So I'll not go with each and every sentence, but you see the suffering from the cancer, male referred in this particular slide, and it is the second most leading diseases in deal with the cancer after the cardiac. Heart disease is the one that is a cancer disease which deals with different types of cancer. There are showing it first is the population of India, 0.2% is also a huge one. We are having more than 100, right? And if you see our percentage, the figure in which is shown in the percentage, they are very much huge one. People. Hence, this is the important one, that why we are need to work more on these such topics, and why we need to understand this topic can be very much understood from these slides. Different types of cancer which the world is suffering. The percentages wise, if you see, the breast cancer is the toppest on the top one, as well as the biliary tract cancer is on the least one. But considering the efficiency of the variations, they can be changed. There can be changes, and all these data are from the world data. That is obviously standard one related to 16. So we need to understand about the immune system. If you consider the immune system, we are very much familiar with the some type of cells which works on the immune on the other body antibody which enters into the body. Now they are called as B cells as well as the T cells. right? So these B cells and T cells work very efficiently to target enemy. Now these enemy are nothing but the viruses, they are the bacteria, they are the other unwanted parts which enter into the cell and which are to be recognized. See, there are some cells, as I told you, T cells and B cells have a responsibility or have the function to recognize those viruses and to recognize those bacteria. And these viruses, if they are not recognized immediately, or in a specific time, they can turn towards the health issues. Now these health issues may be, as we are suffering today, that is the coronaviruses, or may lead towards the cancerous cells, or maybe may lead towards some of the abnormal cells. As some in some of the literature it also says that cancer are the abnormal cells, cancers are the tumor cells, cancer are the unwanted cells. All these terms are just similar to kind of this cancer. So the T cells and the B cells have the response, the unwanted cells which have entered, and to detect the normal cells also. If there are some of the normal cells, they will detect the normal cells and they will produce some of the chemicals which can target towards the antibody. Now if you consider that this is an and these invader cells are being detected by the T cells, and they will bombard some of the chemicals and will dissociate all those unwanted cells which have entered into the body. Now, as I told you, there are some of the habits which are being developed by the body, and these habits can respond after a few years. One of them is smoking, tobacco consumption, etc. All these habits can turn the cell towards a muted cell. They may not able to, the immune system may not able to identify, the immune system may not able to reflect towards those other unwanted cells or unwanted bacteria or viruses which have entered into the body. And they can grow in multiples. They can grow in more amount and form the T cells as well as B cells are not performing their functions. Then what happens? Then obviously what happens is the formation of the unwanted cells. Now the question over here is, why allows to form the unwanted cells? The body allows, if the body has the resistance, these resistance are nothing but these T cells as well as B cells. That may lapse. This lacking is due to the identification of those hazardous cells. The body is not able to identify those invaders. They are not able to identify the good, as well as they are not able to identify the bad ones. That is, they are not performing their functions. Now, generally what happens is these cells have to, just a minute, I will. These cells have to perform and identify by sprinkling or spraying some of the chemical, burn it. But there are some of the times B cells and T cells are not able to activate themselves. Though having an immunity, though having the T cells and B cells, they are not able to identify those cancerous cells because of weak immune system. At this point of time, the immune system is to be made powerful, and this immune system can be made powerful by making stronger or identifying those cancerous cells which are not able to multiply, which are stopped from multiplication. And as we know that they can only be done by T cells as well as B cells by making the immune system powerful. Now, talking about this particular topic, that is about the cancer cells, if you see, in a normally the cell have to balance everything. The cell are supposed to balance everything. If you see, they have to balance, that is the less, sorry, the if we have a more amount, then also more amount of components, more amount of powerful immune system we are talking about. But if there are more number of T cells, extra amount of B cells, then also it may create problem. If we have less amount of cells, that is T cells as well as B cells, they also create the problem. So the balance is to be maintained, that can called as the normal balance. So there will be some stage where excess as well as the less amount, that is restoring of the baseline, is to be applied. And for that, these diseases are making their cells more powerful. The cancerous cells make their multiplication more rapidly in both these conditions. So we need to know that how these can, and these cells are, if it is in excessive part, they will be generating more cancers, they will be having more number of diseases. If it is an insufficient amount of elements or chemicals present, then also there are different types of diseases. So this is a normal tumor cell. But before talking about the cancer cell, we need to cancer mechanism. In fact, we need to understand the process which is very beautifully processed by in our body itself, that what we refer as apoptosis. This apoptosis is a programming. It is nothing but a programming. Now, how this program, what programming and what about is the programming is, there is a mechanism in our body, very beautiful mechanism, that the cell can have a specific time for living, and after that time the cell have to die. It is programmed in its whole system, that is called as programmed cell death. Generally there is a reference which says that a single cell is being living for 90 days. After 90 days, the cell have to regenerate, or it has to die through the process of what we call it is the phagocytosis, and the mechanism which we'll understand in this apoptosis. So this apoptosis is nothing but the breakdown or the programmed cell death, PCD. So this programmed cell death is always being important with reference to the cancer. Now you just understand that if we don't have a program which the cell has to divide, as well as the program that is the growth of the cell, as well as the program where the cell have to die, in both the cases, the cell have to perform very much neutrally. The cell cannot perform excessively, as well as the cell cannot perform inefficiently also. This apoptosis plays a very crucial role in the formation of the diseases. Now there are few slides over here, but I'll not go through each and every slide. I'll run on a quicker basis and understand the basic theme of this apoptosis. This apoptosis works for the cell death. Now there are many parts in our body during the formation of the cell. When in the womb itself, that is, there are few elements. For example, the web I have shown you in the slide also. There are the structures which are formed during the formation of the baby in the womb itself. There are some unwanted cells. These cells are present earlier, initially, but at one stage they will be dying. That is, the size or the web between the two fingers is not allowed genetically in the human beings, if we talk about the human beings, but in other animals they are allowed. So if you see, there are webs present during the formation of the fingers, but as the fingers are formed, the webs between the two fingers or the cells between the two fingers are not present. Hence, this is the mechanism, what we call it as a apoptosis, that is programmed cell death. The cell have to die at a specific time. It is considered to be the term which is called as a apoptosis. The mechanism programmed death is starting with different, we can say that is, there are two processes. I will quickly go on towards those two processes, that is intrinsic process as well as extrinsic process. Now, both these processes works on the specific element or works for activating the specific element or compound. This compound is called as caspase. Caspase is nothing but cysteine aspartic proteases. Cysteine aspartic proteases. Now, these caspases are to be activated. Now, as soon as they are activated, there is a sequence, or we can say there is a different types of series of caspases or family of the caspases which works alternatively or which works subsequently, consecutively. So these caspases may be caspase 3, caspase 8, caspase 9, likewise. So all these caspases works for apoptosis process. Now, cysteine aspartic protease. Now, if you consider the last term, that is protease, that there are two terms or two enzymes which are to be made activated: proteases as well as nucleases. You just remember these two terms, you will able to understand the whole mechanism of apoptosis. Protease is the breakdown of the protein, right? If the proteins are broken down in a cell, you just imagine if the proteins are broken down, what amino acids are broken, preparing the proteins, right? If these proteins are broken down, they will not able to perform any of the enzymatic functions. Hence, if the proteins are broken down, the cell will not able to survive for more time. The second important enzyme which is needed to be activated for apoptosis is the nucleases. These nucleases works for breakdown of the genetic material, that is of the nucleus. What is present in the nucleus? The genetic material, the genetic codes, DNA, RNA, all these are to be broken down. This is done by the second important enzyme, that is the nuclease. So caspase is cysteine aspartic protease, which works targetedly on the protease enzyme that is on for breaking of the proteins. But it also activates the nuclease, which helps in breaking of the nuclease genetic material. Hence, both the important fundamentals of the cell, that is protein as well as nucleus, are being distorted or damaged by these two enzymes, that is protease and the nuclease enzyme. So the apoptosis purpose is to activate the caspase, that is the term which is called a cysteine aspartic protein. So I'll not go through all these slides as I told you. Let's go two important points which I need to make you understand. That is two pathways which are relevant or which are to be understood for the apoptosis process, that is intrinsic pathway and the extrinsic pathway. Both these pathways are very important as far as the whole cancer is to be understood, because if you just imagine, if we don't have a mechanism of apoptosis, that is cell is not able to die, that means the cell is growing and growing. Now there will be the case that the function of the cell will be on the path of dysfunctioning. This dysfunctioning will lead to a mutation, but the cell is not programmed to die. There is no apoptosis. Hence, the cell will be growing and growing. No death is allowed. Hence, this mechanism, apoptosis, is more relevant towards the cancer cells, right? So two pathways which the apoptosis process is following, that is intrinsic pathway and the extrinsic pathway. Intrinsic pathway is, the word itself says that it happens within the cell itself, that is from the internal factors only, and the second one, that is the extrinsic pathway. Extrinsic pathway is some of the external sources which are inhibiting or which are interfering the process of the cell. Hence, external, from the external source, that is in extrinsic pathway. Internal, that is within the cell itself, that is intrinsic apoptotic pathway. Now, the apoptosis triggered by various stimuli from the outside or the inside. Now, I have listed out some of the things in general of both these pathways, intrinsic as well as extrinsic, which are need to be understood. That is ligation of the cell surface receptor. We know that on the cell surface, there are certain kinds of receptors which catch hold of specific ligands, right? And they are to be binded very much particularly, very much specifically. Second is the DNA damage. There are some of the cells which may find damage in their DNA and which cannot be repaired, or which can be repaired by the other mechanism that is called as DNA repair mechanisms. Treatment of the cytotoxic drug, or we can also by through the lack of survival signals. There may be no signal. Contradictory to the cell signal, cell may not receive any of the signal, or if it is receiving a bad signal, or it is receiving some of the anti-signals by the development of the developmental death signaling. So there may be some of these criterias where the apoptosis is started or initiated. Now, as I told you, I'll skip towards the two important pathways because I think I have crossed the time duration, but I'll continue within 5 to 10 minutes. Right, intrinsic pathway as well as extrinsic pathway. These intrinsic as well as extrinsic pathway works in its own way. Intrinsic pathway, as I told you, there is a mechanism which is starting from the cell itself, from the within the cell, intrinsic, internally, and this internal mechanism starts with the damage of the DNA. The cell may have found itself a damage within itself, that is maybe in the DNA or maybe in any of the other enzyme which has excessively increased, any of the cases. Now, for example, I have written in this slide, that is due to some of the protein, that is maybe serine, maybe threonine kinase, which is activated by the specific gene. Now, as it is activated by the specific gene, they have found itself to be a damage in the DNA. This DNA damage can be formed or can be repaired in some of the mechanical way, or it can form a tumor, or it can be initiating the tumor suppressor gene. Now, the word itself, tumor suppressor gene, is itself says that tumor is being suppressed. Hence, apoptosis is being formed, or it is being prevailed. So tumor suppressor gene, especially here, I have referred one of the examples, that is P53. P53 gene is so crucial that if it is not getting activated, there are chances that cancer will be generated, or the cancer will be formed excessively. So the P53 tumor suppressor gene acts as an initiating phase in the intrinsic pathway. Now, in many literatures also, intrinsic pathway is running towards the it's called as a mitochondrial pathway also. Mitochondrial intrinsic pathway, it is also referred in some of the literatures. So mitochondria plays a crucial role in this intrinsic pathway, in mitochondrial intrinsic pathway. There is a signal which is being received from, as I told you, example, DNA damage. There is a signal which is received by the cytoplasm, and in the cytoplasm there are some of the enzymes and proteins which are available, which are waiting for those types of signals. Now, here there may be BAX type of enzyme in the intrinsic pathway, or maybe BCL2, another type of enzyme or protein which is helping in the apoptosis mechanism. So this enters into the mitochondrial membrane. As soon as it enters into the mitochondrial membrane, the BAX protein or the BAX enzyme, they are supposed to release some of the chemicals, and out of these chemicals, for example, calcium, H plus ion, out of these chemicals, cytochrome C is one of the important which is to be released. Now, this cytochrome C is considered to be the important step for the process of apoptosis. Cytochrome C, because eventually will be activating one of the other factor which helps in the apoptosis, and this factor is APAF, that is apoptotic protein activator factor. This apoptotic APAF, it is also referred as APAF in short. APAF is one of the crucial compound which is very much known for activating the caspase, which is activating the caspase 9 over here in this intrinsic pathway. Caspase 9 eventually activates the caspase 3. The caspase 3. So we have a Bax enzyme. I think you all may be remembering about the mitochondrial membrane. There are porins available in the mitochondrial membrane. So through the porins or through these spaces, the Bax BACL2 will be signaling those mitochondrial membrane that they have to release the cytochrome C. Now for the release of cytochrome C, they will be, as soon as it is released, the apoptotic protein activator factor will be activating the caspase 9, and this will be activating caspase 3, and which is considered to be the final step. As soon as the caspase 3 is activated, the whole apoptosis, that is the whole damage of the cell, is now being confirmed, now in a general sense. If you see, we all have seen a cell in a circular form, right? When the apoptosis starts, this mechanism is the cell will be forming in a bulby shape, or it is called as a blebbing. The cell forms like a finger-like projection. Now this bulbing is the initiation of the process of the apoptosis, and the cell has confirmed itself that it is to go for the death pathway. Hence, the apoptosis in the intrinsic pathway conforms itself with the DNA damage. Bax will be allowed to enter into the mitochondrial membrane. This cytochrome C will be released due to the entry of the Bax. Cytochrome C, as it is released, APAF will be activated, which is one of the factor, which is a protein, apoptotic factor, which activates the caspase 9. Caspase 9 activates caspase 3, which is considered to be the final apoptosis. So this is the whole mechanism in the graphical form pathway. So if you consider both the factors, that is intrinsic extrinsic factor, they are to activate the caspase 3, or they are to activate the caspase 8. Now here, if you see the extrinsic pathway, there will be some of the external source. As I told you, there will be some of the external factors for any of the other chemicals which are not to be tolerated by these cells, and they are not able to identify those chemicals. They are not able to resist those chemicals. For that, there are TNFR which are present inside the membrane, or present on the membrane of this cell surface. So TNFR, that is tumor necrosis factor. These tumor necrosis factor will be helping for the process of apoptosis. And generally, there are fast enzyme or fast proteins which are considered to be one of the important or initial phase of the extrinsic pathway. Now, as there are TNFR which are present, these TNFR will be binding with a ligand. These ligands are nothing but TNF alpha, maybe TNF, that is tumor necrosis factor receptor, and they will be binding with the TNF ligand, that is maybe TNF alpha, for example. As soon as they bind with the ligand, here inside the membrane, there will be the subordinates. These subordinates here is called as death domain. These death domain will be joining with those TNFR, those receptors from the inner side, and these inner side receptors, or the inner part of those receptors, that will be forming the death domain with the combination of death effector domain. Now, as these death domain combines, they are now called as a complex structure, which are referred as death effector domain. Now, eventually, when these death effector domain are being joining with the death domain, they activates caspase 8. Caspase 8, that is nothing but one of the important initiating for or confirmation for the process of apoptosis. This process of apoptosis starts with the conformation or activation of caspase 8, and this caspase 8 once again goes for the caspase 3 activation. 3 activation means there will be once again the blebbing, and these blebbing will be nothing but the finger-like projection, and the cell has decided itself to go on the death pathway. So extrinsic pathway works from the external source, that is TNFR. TNFR is nothing but tumor necrosis factor receptor, and it activates, it binds with the ligand, that is tumor necrosis alpha maybe, as well as the FAS, and it forms the death domain. This death domain combines with another domains inside the membrane that is called as death effector domain, and these will activate the caspase 8. This caspase 8 will finally activate the caspase 3, which is taking the responsibility for the process of apoptosis. So I think I have taken much more time in the process of respiration as well as apoptosis. So I'll Rupesh bhai, are you there? I can continue for a couple of minutes.
Yes, sir.
I think complete this
Yes,
as a
sir.
whole,
Yes,
right?
sir.
Yeah, thank you very much. So if you talk about the apoptosis, that is important as a part of the cancerous, not to generate the cancer cells. Here, if you see, there are list of genes which are available for the whole mechanism of the cancer. There are BCL2, as I told you earlier. There are P53 genes. There are RAS genes. All these types of genes, MYC genes. MYC genes are major important function is to have an important involvement in the protein interactions, that is protein to protein interactions, as well as they have a ability to perform a dysfunction also, that is not a normal function but to have more multiplication of dysfunctioning cells. So these are some of the crucial genes which can form the cancerous cells. In a very quick time, I will try to go through the tumor suppressor gene. Now, this tumor suppressor gene, as I told you, tumor necrosis genes or tumor necrosis factors. Now, these tumor suppressor genes are able to inhibit the growth of the cells. Normally, they are not they are not forming the tumor cells. They are suppressing the cells which are able to form the tumors. Hence, they will be directly associated towards the proteins, those proteins which are changing their functions. These proteins can, at a later stage of time, can form a bad protein or a dysfunctional protein. Hence, they will allow the cancer cells. So for that, the cell has the mechanism to stop those bad proteins with the help of tumor suppressor genes. In any cases, whenever the cancer forms, it is nothing but the mutations. Mutation in these genes result in the cells that have no longer the normal functions of the growth as well as of the divisions of the cells. Right. It means that the characteristic is not expressed unless both the copies of the normal genes are muted. If you see, the growth of the cell is also important, that is the cell division is also important, as well as the apoptosis mechanism is also important. The example which I have written in these two in the last sentence, that is, we need we need an accelerator in the in any of the vehicle, as well as we need the brakes also. That is, we need to go in a very higher speed also, but also we need to go on a safer speed also. So that is the two purpose for these tumor suppressor genes, which deals the growth as well as of the division. If you consider the basic tumor suppression gene mechanism, I have shown you this particular figure that deals with one of the important cancer ability of these tumor suppressor gene. Now, during the formation of the child in the womb, if you see, there are specific genes which are to be, if they are not activated, they form a highly dangerous diseases. If you see both the eyes of this particular child, they are having differences. One is normal one, the another one is a affected one, or which is having some sort of disease. This disease, you will not believe, it is a cancer. It is referred because of inability or because of dysfunctioning of the tumor suppressor gene, one of the tumor suppressor gene. Now for that, the term is referred as retinoblastoma. The eye which you saw in the different, having the difference, that is the retinoblastoma, due to one specific mutation in the gene, that is retinoblastoma. This tumor suppressor gene was not able to go for an efficient gene and mutated itself and led towards the development of this particular retinoblastoma. So in all, if you see, consider the whole cancer. This cancer, I've just partially completed this particular topic of cancer along with the apoptosis, but one of the half part of this cancer, which deals with the oncogenes, is still to be completed. Oncogenes are the genes which are one of the responsible for the cancerous cells, that is the oncogenes, and these oncogenes are formed due to the mutation. I have listed over here some of them, that is MYC gene, RAS gene, cis gene. All these are some of the genes which are associated with the cancerous cells, or which are helping in building up the cancerous cells. So if you consider the oncogenes, they are the oncogenes as well as which are formed from the oncogenes. We need the proteins. These proteins are formed. These proteins, if we are behaving normally, they forms normal functions, performs normal functions. If you see, proteins go for growth factors, development of growth hormone receptors. They also go for GTPase. They also go for transcription factors. All these factors are important if the protein are behaving normally. But if you see on the left hand side, the proto-oncogenes, if they are not behaving normally, if they are being muted, they form the oncogenes. They form the oncogenes. These oncogenes are nothing but the muted one. They have made changes in themselves and produced genes which are also functioning but not normally. These genes are called as oncogenes, and these oncogenes also produces proteins, and these oncogenes which produces proteins are called as oncoproteins. Oncoproteins functions normally, but they are having an abnormal mutants, which is called, for example, if you see here, mutant MYC, that is MYC gene, which is responsible for transcription, multiplication of the genes. That is MYC gene, during their time, they will be not transcripting, but for the bad proteins. Will be transcripting themselves for the bad proteins. Hence, the function will be very much overexpressed. They will be overexpressed. We have oncogenes which produces oncoproteins, but these oncoproteins are not the normal proteins. Hence, they are overexpressing any of the genes. Here in this case, I have written an example of MYC gene, which is responsible for transcription. And these overexpression lead towards the amplification, more and more number of amplified gene which are not normal gene. And hence gene amplification process starts, which leads towards the cancer cells. These cancer cells are very much dangerous in terms of the functioning of that particular organ on which they have found their substratum. So in this one single slide, I have tried to show the oncogene as well as the difference between the proto-oncogenes and oncogenes. If the cells are having the proto-oncogenes, they are considered to be the normal one, the best award citizenship, as I told you. These proto-oncogenes, whenever they are muted, they will be not normal. They will behave normally, but they will not function normally. They will be having different types of overexpression of different genes, which I shown you in the earlier slides. All those genes, if they overexpress themselves, they are functioning in a different pathway, they lead towards the cancer cells. Hence, it is very important to understand the proteins, to understand the abnormal proteins for its functioning, right? So almost all these normal cells can have the ability, if there is no apoptosis, if there is more growth of those abnormal cells, they can lead towards the cancer cells, right? So I will stop over here, and I think I have majority of the part, that is apoptosis, as well as this tumor suppressor gene, I have tried to cover, and I also tried to cover the oncogenes, but the metabolism of these are still being remained. So I think in the next session, if the time permits, I will be trying to cover this particular thing. So I thank all the organizers as well as the team of the whole Guru Govind's University for inviting me and for making me to able to deliver this particular topic of respiration as well as medicine. So thank you very much. Thank you all for paying the kind attention towards the topic. Thank you. Thank you all.
Thank you, Suhas sir, for delivering such a nice and detailed lecture. Taking 2 hour lecture is always not easy, but you delivered it very nicely. And even though you completed respiration, you went on to take uh, the second thing of cancer part, and even in the cancer, you took almost all the parts. So thank you again, Suhas sir, for delivering such a nice lecture. And if possible, we would also like to arrange one of the lecture on cancer, the metabolism part,
Definitely.
whenever you will
Definitely.
permit us to arrange the lecture. So because
The
it is
important
one,
part I want to cover, that I have completed the apoptosis as well as the tumor suppressor gene. Some of the part I have also completed, but the major part of oncogene is to be once again, I think it is to be continued as a part of metabolic activity. How the cell cancer cell perform the glycolysis, because they also does all those pathways, they also go for all these mechanisms, glycolysis and all. So those things are a little bit of in the remaining part, but let's see in the next few days.
yeah, sure, sir. So thank you for presenting such a nice lecture. And now I would go towards our student representative, Saif and Jaimin. Please turn on your camera and mic. Are you alive, both of you? Hello,
Yes, sir.
Saif, turn on your camera. Jaimin.
Yes, sir.
Okay, Saif. How was the lecture? Please provide your review, or if you want
Yes,
to
sir.
ask something to Suhas sir.
Yes, sir. શ્રી ગોવિંદ ગુરુ યુનિવર્સિટી ગોધરાના વાઇસ ચાન્સેલર પ્રોફેસર પ્રતાપસિંહ ચૌહાણ સર અને પ્રોજેક્ટ કોઓર્ડિનેટર ડોક્ટર સોની સરના માર્ગદર્શનમાં નેટ જીસેટના વર્ગો ચાલી રહ્યા છે, જેમાં આજના વક્તા શ્રી ડોક્ટર સુહાસ વ્યાસ સરે ખૂબ સરસ રેસ્પિરેશનમાં ગ્લાયકોલિસિસ, ક્રેબ્સ સાયકલ એન્ડ ઇટીસી અને કેન્સરનો થોડોક ભાગ પોતાનો ટોપિક રજૂ કર્યો, જે અમને નેટ જીસેટમાં તો ખરું જ, પણ જેને બીએસસીમાં પણ આ ટોપિક આવતો હોય તેને સમજવામાં આ માધ્યમથી ખૂબ જ ફાયદો થશે. અને સાથે સાથે લાઈફ સાયન્સના જીપીએસસી યુપીએસસી જેવી એક્ઝામમાં ઓપ્શનલ સબ્જેક્ટમાં પણ સરળતા રહેશે. ડોક્ટર વ્યાસ સરે ખૂબ સરસ અને સરળ રીતે ટોપિક રજૂ કર્યો. થેન્ક યુ સો મચ સર, એન્ડ થેન્ક અ લોટ સર. એન્ડ શ્રી ગોવિંદ ગુરુ યુનિવર્સિટીના વાઇસ ચાન્સેલર પ્રોફેસર પ્રતાપસિંહ ચૌહાણ સરના માર્ગદર્શનમાં જે નેટ જીસેટના વર્ગો કે જે ઓનલાઈન માધ્યમથી ચાલી રહ્યા છે, તે માટે હું પઠાણ સૈફ ખાન, સ્ટુડન્ટ બીએસસી સેમ 3 સેટ પીટી આર્ટ્સ એન્ડ સાયન્સ કોલેજ ગોધરા થી વાઇસ ચાન્સેલર પ્રોફેસર પ્રતાપસિંહ ચૌહાણ સર, જે પ્રોજેક્ટ મોટિવેટર પર છે, અને પ્રોજેક્ટ કોઓર્ડિનેટર એસજીજીયુના મીડિયા કન્વીનર ડોક્ટર અજય સોની સર, અને લાઈફ સાયન્સના કોઓર્ડિનેટર ડોક્ટર રૂપેશ નાકર સર એન્ડ મિસ ફાલ્ગુની પરમાર મેમ. તો ખૂબ આભારી છું જેમણે મને તક આપી. નેટ જીસેટના ઓનલાઈન વર્ગોમાં પીએચડી એન્ટ્રન્સમાં ઘણા ઉપયોગી થશે, અને સાથે સાથે જીપીએસસી યુપીએસસી જેવી કોમ્પિટિટિવ એક્ઝામમાં પણ ઉપયોગી થશે. અને ઘણા ટોપિક અમારે બીએસસીમાં પણ આવતા હોય, અમને સવિસ્તાર સમજવામાં આ વર્ગો ઘણા ઉપયોગી થશે. ફરીથી આપણી નેટ જીસેટની ટીમને ખૂબ ખૂબ આભાર, અને પીએચડીના ગાઈડન્સશીપમાં પણ નાકર સર જેવા દિગ્ગજોની ટીમ અને ગાઈડન્સ કરશે, જેનાથી હું ખૂબ આનંદિત છું. અને કોંગ્રેચ્યુલેશન સર. થેન્ક યુ સર.
Thank you, Saif, for your very kind words, and we wish you very best for the bright future. And now I would ask Waghela Jaimin to have a review.
હેલો
Jaimin bhai,
સર.
ji.
હા સર, શ્રી ગુરુ યુનિવર્સિટીના વાઇસ ચાન્સેલર પ્રતાપ સરના માર્ગદર્શન હેઠળ જીસેટ નેટ સેટની પ્રિપરેશન માટે આ વેબિનાર યોજે ખૂબ ખૂબ આભાર. અને અમને આ વેબિનાર વિશે અમને થોડી વધારે માહિતી મળી એના માટે ખૂબ ખૂબ આભાર. અને અમે આગળ શું કહેવાય પ્રિપરેશન માટે અમને આ ટોપિક વિશે વધુ માહિતી મળી એના માટે ખૂબ ખૂબ આભાર સર.
Thank you, Jaimin and Saif, for your active presence in the lecture, and hope you have enjoyed the lecture throughout the session, as sir was presenting very nicely. And Falguni madam has depicted all the points which were in the form of summary, which were undertaken by Suhas sir. So over to you, Falguni madam. Please go for depicting points as well as vote of thanks.
Thank you, sir. Good afternoon, everyone. I would like to depict some key points from today's lecture. Suhas Vyas, sir, and topic respiration and cancer. In respiration, glycolysis, Krebs cycle, and etc. were the major part to be covered. Respirations is of two types: anaerobic respiration and aerobic respiration. In both in anaerobic and aerobic respiration, glycolysis is a common step. And in anaerobic respiration, two molecules of pyruvate generated during glycolysis are will go in the fermentation or methyl alcohol and carbon dioxide will be formed, or lactic acid will be formed. In aerobic respiration, three parts are main: glycolysis, which occur in cytoplasm of and etc. Electron transport chain occurs in the inner membrane of mitochondria. Another is oxidative phosphorylation, which combines the electron transport chain and Kemi Osmosis. Glycolysis has two phase: energy production and energy is consumed, and second is in which energy produced. First phase is of 4 reactions, and the second phase is of another 6 reactions. Total 6 ATPs are produced of total 8 steps, and NADH and FADH and ATP major are produced during citric acid cycle. Third is electron transport chain, which occurs in inner mitochondrial membrane, inner part of mitochondria, and it contains 4 complexes and ATP synthase enzyme. Energy calculation per molecules of glucose, out of 1 glucose, total 32 ATPs are produced during aerobic respiration. Next topic was cancer, in which sir discussed a statistic of the cancer in India, particularly which type of cancer and how many patients are there each type of cancer. Then immune system, after that apoptosis, 2 types of apoptosis, intrinsic, 2 pathways of apoptosis, intrinsic and extrinsic pathway. Then cell biological approach of cancer, in which genes associated with cancer like BCL, BLM, RAS, then P53, XP, etc. Then tumor suppressor genes and oncogenes. Thank you, sir, for delivering a very detailed lecture on respiration and cancer as well. Now, I would like to express my vote of thanks. Today we completed our 21st lecture of series 2 in life science NET GSET PhD program preparation, exam preparation program. On behalf of NET GSET PhD MSc Life Science Department, Shri Govind Guru, I take this to propose vote of thanks to all those who have directly or indirectly in our today's session. I would like to extend very hearty vote of thanks to our today's esteemed speaker, Dr. Suhas Vyas, for taking out the valuable time of his busy schedule and delivering very detailed lecture on respiration and cancer. We are very lucky to have you with us. Thank you, sir, for delivering second lecture. I wish to express my gratitude to our Honorable Vice Chancellor, Dr. Pratap Singh Chauhan, who is project motivator of this program, and thank you for motivating us. Dr. Respected in charge registrar Dr. Anil Solanki sir for providing us his valuable guidance. Dr. Ajay Soni, coordinator of NET GSET PhD MSc Entrance Exam Preparation Unit of Sri Govind Guru University, and also initiator of this program, and for always inspiring us to work more and more and encouraging us. Thank you to all EC members, AC members, principal of all science colleges, and professors of all life science departments. I would also like to present my vote of thanks to our today's guest, Dr. Kanak Lata Madam, for providing best wishes and motivations, and for providing her blessings, and for appreciating our work. Thanks to our today's student representatives, Jamin Wagela and Khan Pathan from SLET Arts and Science College, Godhra, for sharing your reviews and for being connected with us throughout the session. At last, special thanks to Mr. Jagrut Pandya, Milan Shah and their team for providing technical support throughout the program. And thank you, Dr. Rupesh Nakar sir, who is coordinator of NET GSET PhD entrance exam preparation of Life Science Department, conducting this program, and all the arrangements. And thanks to all the students watching us live on YouTube and Facebook. And thank you each and everyone. Thank you all the dignitaries present over here. And congratulations to Nakar sir for getting PhD guideship in botany, and I am always motivated by you, sir. Thank you, sir. Over
Okay.
to you, Nakar sir.
Thank you, Falguni madam, for such kind words and nice words, and we are also thankful to you for your very nice work in all the sessions wherever you worked. You worked with very precise pattern, and even today's seminar you depicted most of the points. Thank you again, Falguni madam, for such a nice vote of thanks. Thank you. So I think with that we have concluded our 21st lecture, where Dr. Suhas Vyas delivered a lecture, informative lecture on respiration and cancer. And the next portion, I think we will arrange in short span of time, whenever time will permit. So with that, I think I should conclude the meeting. And Suhas sir, thank you again for providing your very precious time to us and to
Thank
all
you.
the
Thank you.
students. And
Thank
you
you
were
very
very
much.
nice to us. Thank you so much, and hoping the same response from you always, sir.
Thank you. Thank you. It's obviously sure done.
Thank you, sir. And thank you, Jagrut Bhai and whole team. And now I think best wishes to all of you. Have a great day. And the day after tomorrow, we will meet again in the session, the same time, 1 to 3. The topic will be different. Till then, stay tuned. Have a great day. Bye bye, all.
Thank you.
Thank you, sir. How, Joe?
Yes, sir.